Okay, so good morning, good afternoon, good evening, everybody. Hope everybody can see us and hear us. My name is Charu Kaushik, and I am the Scientific Director of the Institute of Infection and Immunity at the Canadian Institutes of Health Research and a co-chair of the GLOPIDAR. And I welcome my co-chair, Yazdin Spana, who is the chair of the GLOPIDAR, as well as the director of INSERM Reacting Consortium and the INSERM Institute of Infectious Disease, Microbiology and Immunology. On behalf of GLOPIDAR, I welcome you all to the COVID-19 Research Synergies meeting. And today we're starting off this series of meetings with the vaccine session. So Yazdin, I'll ask you to say a few words about GLOPIDAR.
Thank you, Charu. So good afternoon, good morning, good night to everyone, because we have people from all around the world. So I'm just going to say very few words on the GLOPIDAR, which is an international network of research funding organization that was launched in 2013 to facilitate, accelerate, and deepen collaboration between research funders during emerging diseases, with the idea of investing in strengthening global research preparedness between crises and in mobilizing resources to respond rapidly and effectively to these infectious diseases. Although now we can see that there is no between crisis, basically. We have all the time in crisis. So the G20 recognized GLOPIDAR, the G7 welcomed its action, and the EC financed the GLOPIDAR Secretariat through the Foundation Merieux and University of Oxford. Next slide. So today we have more than 29 members and 2 observers from around the world. As you can see, all the countries that are involved as funders in GLOPIDAR. Next slide. So regarding the response to COVID-19, we have members, observers, and stakeholders that were mobilized in the response. I'm not going to detail everything, but we first collected information on members and existing research activities. We had close collaboration since the beginning with the WHO, and particularly the WHO blueprint, and in particular in the organization of the roadmap that was organized in Geneva in February 2020. In launching emergency calls, and what we really tried to do was to coordinate funders as much as we can to try to optimize resources to avoid duplication and to cover priorities listed in the research and development roadmap of the WHO. Next slide. And to finish, because I'm 2 minutes, I just, at that time, we also decided that it would be good to have all the researchers together after a few months to talk about what they have been done. Thanks a lot. Back to you, Charu, and I wanted to particularly thank Charu and everyone for all the great work for organizing these sessions.
Thank you very much, Yazdin. So with that, I'll do a few housekeeping notes and then welcome the co-chairs for this session. So just to let all the participants know that we will be taking audience questions during the panel discussions, and to particularly point out to you that if you want the questions and answers or questions to be considered, please use the Q&A box that is there at the bottom right next to the chat box. The chat box might have a lot more chatting and other discussions going on. So we are going to focus only on the Q&A session rather than the chat box. So feel free to use the chat box for other discussion points. I also want to let everybody know that this meeting is being recorded and live streamed on YouTube. And then with that, I would like to welcome the co-chairs of this session, Dr. Anita Zaidi, and substituting for Dr. Melanie Saville today is Dr. Deborah Yaski. I would like to express regret on Melanie's behalf. Part of the issue of having amazingly high profile co-chairs is that they're actually very much involved in the response to COVID. So Melanie, unfortunately, yesterday had to tell us that she was pulled out for an emergency meeting this morning. She will be joining us on the final session on the 23rd. And I'd like to thank Deborah for stepping in at the last minute and able to substitute for Melanie. Deborah Yeske is the head of regulatory affairs North America of CEPI. Before this, she was serving in similar positions, head of director of regulatory and quality affairs division in BARDA and in the US Department of Health and Human Services. So welcome, Deborah, and thank you so much for doing this at the last minute. And Dr. Anita Zaidi, who is a physician and the director of vaccine development, surveillance, and enteric and diarrheal disease program at BMGF. Anita has worked extensively on vaccine development in LMIC, surveillance, and has done a significant amount of work in this area. I'd like to thank Anita and Melanie, who have worked tirelessly with the team to put together an amazing program today. And finally, I'd like to acknowledge the team who's been working behind the scenes. My colleagues from CIHR, Genevieve Boilly-LaRouche, Deborah Cursigera, and Danielle Vitali, who are handling everything from behind the scenes. So thank you very much. And with that, I'll hand it over to you, Anita.
Thank you, Charu, and greetings and welcome everyone to this session 1 of the GLOPEDAR meeting, which appropriately is on vaccines. And I hope that we will have a fantastic discussion and presentations today. Vaccines are our best hope for ending this pandemic. I want to thank Charu, you and your team at GLOPEDAR for putting together the program, and I look forward to discussions as we go through the program. To set the scene, we will start the program by an overview of where we are today in SARS-CoV-2 vaccine development. And as a scientific community, this is unprecedented progress that has been made in 6 months in tackling this pandemic. What normally you would see happening in 5 to 7 years, we've seen happen in 5 to 6 months. There is huge scientific engagement across the world, many people, many institutions involved, but it is appropriate to start the program today with a representative of CEPI, the Coalition for Epidemic Preparedness and Innovation, Dr. Deborah Yeske, to give the opening talk. And for those of you who don't know, CEPI is a young organization. It was founded only in 2017 in the aftermath of the horrible West African Ebola crisis. Co-founders included Norway, UK, EC, Germany, the Wellcome Trust, and the Bill and Melinda Gates Foundation. And who knew that CEPI would be challenged in this way so soon after its founding? Let me hand over to Dr. Deborah Yeske, who is, as you heard Dr. Kaushik say, substituting for Dr. Melanie Savelle at the last minute, in fact, only yesterday, because of this really important meeting that's happening today, which I'll mention later on in the program, which will influence a lot of what we see happening over the next 6 to 12 months. Thank you. Over to you, Deborah.
Thanks, Anita, and thank you to Charu and all of the folks that have put this, and Melanie, to put this great agenda together for today. So I'm just going to take a couple minutes to talk about the scene setting for vaccine development for COVID-19. Next slide. This is just to give this graphic just kind of saw it yesterday for the first time, just jumping in. It's just amazing to see that there are over 200 vaccine candidates, starting for COVID-19. And as we can see from here is that, the top developers by region, geographic location, we see North America, Europe, China, and Asia are leading the pack here as far as vaccine development. Next slide. This is just a snapshot again of the pipeline that of the vaccines that are in the clinic. And it was, even though it says the date of today, I got this yesterday, so it might be out of date even as of today, but it just gives you, and I know it's hard to see, but it just gives you a snapshot of those vaccines that are in the clinic. And then just to note, and I'll get into it a little bit later, that the pink shaded ones are right now in what we call the COVAX portfolio. And I'll talk about that a little bit later. Next slide. Again, just scene setting and just to for us to get our arms around the vaccine development landscape. This graphic just shows that the previous slide were the vaccines that are in clinic now. These are the vaccines which are more than 60 slated to be in the clinic by quarter. And if you look at Q3, we're talking about close to 27 vaccines will enter the clinic in third quarter, which is right around the corner. Next slide. So, as we all know, in this audience, the traditional vaccine landscape and how vaccines get licensed by regulatory authorities, and it's largely sequential. However, for response to COVID-19, we've had to compress timelines and do things in parallel and at risk. And that means, we will talk a little bit about our mantra, speed, scale, and access. This, means that, we are manufacturing at risk, which then puts, there's a financial risk to that as well. Next slide. Just to highlight the different vaccine platforms for COVID-19, and we know these, and they all have different attributes. No one vaccine is perfect and has all the attributes that you would want in one single vaccine platform. For instance, the RNA vaccines are very fast and usually the first to get into the clinic. However, when you look at the scale, they're usually low to medium scale. And as conversely, the recombinant protein vaccines are a little bit slower to get in the clinic, but the scale is higher, and it makes it more attractive for making millions to billions of doses. Next slide. This is just to highlight the CEPI COVID portfolio. We have 9 candidates in our portfolio, and nearly all of them are in the clinic. And again, we are progressing with speed, scale, and access. And it's very important for response to this pandemic to, as I mentioned before, to do things, in parallel so that we can scale up when we're doing things at risk, and to make sure, and this was in CEPI's mission statement, is it's very important to us for equitable access. And then for this in particular, access once we have those vaccine doses and they're approved or licensed to get them to those that need it quickly. Next slide. So just a little bit about the Access to COVID-19 Tools Accelerator, the ACT Accelerator. It has 3 pillars. And so the you know, I'm going to try not to use the word unprecedented, because I think that we all use it, but it really is unprecedented times, and that, you know, this global pandemic requires an all hands on deck, and this ACT Accelerator is a result of that. And we're bringing together through the ACT Accelerator, you know, global stakeholders to respond to and protect the global public health. There are 3 pillars here: the diagnostics, therapeutics, and vaccines. The vaccine pillar is also termed as COVAX, which is co-led by CEPI, Gavi, and WHO. CEPI in particular is the work stream lead for the development, research, development, and manufacturing of vaccines. And then Gavi for the procurement and delivery, and WHO for the policy and allocation. So within the CEPI work stream for the research, development, and manufacturing, we're working hand in glove with all of our stakeholders, including our developers, for an end-to-end process. Next slide. So our goal for the COVAX is what's on the slide there, and it's a lofty goal, but I think in no other time is the world so focused on one goal. And so while it's daunting and lofty, I think you know, we are all striving to meet this goal. Next slide. Again, just to reiterate the COVAX, we have a number of bodies within the COVAX work stream to leverage a unified voice, again, looking at the end-to-end tools to help our developers meet the goal of getting safe and effective vaccines in a very timely manner to get those out to the citizens of the globe. We're working with transparency with our partners. We're looking for innovations, and we're utilizing all of our partners' capacities and capabilities. Next slide. And I think that was my last slide, but just to
So it looks like we're having some technical glitches. Anita, did you want to take over? Are you?
Sure. I think Deborah was just ending, so that I think the timing was not that bad. As you heard from Deborah's slide, we have over 20 candidate products now in the clinic already against SARS-CoV-2 and several starting phase 3 in the next couple of months. There is a very ambitious goal of having 100 million doses for people around the world by 2020, so within the next 6 months, and 2 billion doses by 2021. Now, that's easier said than done. And there are many issues that will need to be tackled before we can get there. And some of them we are going to be trying to cover today in the presentations. We have an hour for the first section, which is on correlates of protection and safety, two really important areas in vaccine development. We have 4 speakers. All of the speakers are requested to keep to the 10 minutes that they are allotted, and we will take questions and answers together at the end. And if you would put the questions and answers in the Q&A section, not the chat section, that would be very helpful. So with that, let me introduce the first speaker, Dr. Volker Goetz from the University of Saskatchewan. Dr. Goetz is the director and CEO of the Vaccine and Infectious Disease Organization, International Vaccine Center, VIDO Intervac for short, which is located at the University of Saskatchewan in Saskatoon, Canada. So Dr. Goetz, all yours.
Good morning. Thank you very much. Can you hear me okay?
Yes, we can hear you.
All right, well, thank you for the invitation. So just a brief introduction to VIDO Intervac for those that are not familiar with it. We're one of Canada's largest research organization focused on infectious diseases and vaccines for both animals and humans, have commercialized a number of vaccines, as you can see here, spun off companies and hold the university's largest patent portfolio. Next slide, please. But most importantly for COVID-19, we led Canada's response. We were the first in Canada to isolate the virus. This was in partnership with Sunnybrook in Ontario and the National Microbiology Lab. We were the first in the country to have an animal model established, and that was the ferret model, but now we also have the hamster model established, and ourselves are working on a vaccine that I will talk a little bit later. As you can see, we're of course member of these expert groups, as many of us are. But then the last one is really the most important on this slide here. We operate one of the largest high containment labs in North America, and I'll show that on the next slide. And we have now received well over 100 requests from all kind of industries, small, large, international, national, academic collaborators, and so on, to help them in their work. And we're using our animal models for the preclinical work for these candidates. Next slide, please. So this is the Intervac facility. As I mentioned, it's one of the largest in the world. The point here of this is that we can house thousands of hamsters and ferrets in this facility. So we can do many, many trials in parallel. And this is what I was mentioning before. We're happy to assist others in their preclinical work using our animal models. And we're also establishing up here in this part of the building a pilot scale manufacturing facility, which will come on stream next year and will hopefully help with production of the vaccine. Next slide, please. Just to acknowledge, there's lots of people involved in the work, and it's mainly led by Daryl Falzerano, one of our scientists here. So I thought it was important that we acknowledge him and the work he's doing. Next slide, please. So in terms of the ferret and hamster model, we established that in March already. So as many other around the world are seeing, ferrets overall show a very mild clinical disease. However, they're a good model to assess viral replication in the respiratory tract, in particular the upper respiratory tract. The hamster is a little bit more clinically, a little bit more severe. And again, allows us to assess replication in the upper respiratory tract as a readout for vaccine efficacy. Next slide, please. So this is just showing here, if you click on it again, maybe that picture on the left side didn't come through, that's fine. So this is just an overview of the initial experiment. We've done now several experiments in ferrets, and the results are fairly consistent. So going in intranasally with a challenge. Next slide, please. And you can see that we can isolate the virus back from the nasal wash, to some extent also from the oral wash. There is very little in other mucosal sites. And then on the bottom left here, you can clearly see that there is large quantities of virus that can be found in the nasal turbinate, so the upper respiratory tract. And typically within the first 4 to 5 days is when we see the peak of those. Next slide, please. When we look at the pathology, it's consistent with what you would see with pneumonia. We're currently doing some work characterizing this in more detail, but obviously there is pathology in these animals and gross lesions that you macroscopically can assess and so on. So indicative of an pneumonia happening in these animals. Next slide, please. For the hamster, it's a little bit more severe in that some of these hamsters, depending on the dose, are getting very, very sick. Their weight loss can be dramatic in these animals. We also find an age difference, as you can see on the top left there. Older animals seem to be more susceptible than younger animals. And then as you can see in the bottom right, there's large amounts of virus that can be re-isolated again in the upper respiratory tract from these animals, so by doing nasal washes. Next slide, please. So now in terms of our own vaccine development, if you can click again, please. We're working like many others, and as we heard in the introduction, on a subunit vaccine candidate. We have various constructs and testing right now. Our main focus is on an optimized S1 at the moment, but we're also working on the RBD and so on. And so what is key for us is, of course, like many others are doing, looking at the conformational structure of the antigen, especially the trimer formation. But then where we are putting a lot of effort in is identifying the proper adjuvant for our vaccine. I will show you the data later on, but this seems to be critical in determining vaccine efficacy. If you don't have a good Th1 inducer, we believe that the vaccines will not be effective. And so we're using a proprietary adjuvant that was developed as part of a larger Grand Challenges in Global Health program funded by the Bill and Melinda Gates Foundation. We've tested this now with a number of our glycoproteins in here for COVID-19. It seems to be highly effective, and I'll show you the data. We're expressing the protein in 2 mammalian cells, HAC293s, as well as CHOs, and the reason for that is that the protein has a number of glycosylation sites. There's 13 of them that are critical, and so we think it's important to assess this by using different mammalian cells that allow different glycosylation patterns to see if any of that relates to differences in immunogenicity. Next slide, please. And so here's where we are at the moment. We're currently making GMP material, both of our adjuvant as well as our constructs. Engineering batches are already available and being used for safety testing. So you can see we're right now here in the safety studies, and then have scheduled our first human clinical trials in October of this year. Next slide, please. So this is just showing you now here 2 vaccine candidates using 2 different adjuvants. So there is the adjuvant that I mentioned, our proprietary adjuvant, which is a good Th1 inducer. And then for comparison, we're using a commercial adjuvant that is being now used by many of the vaccine developers, that is provided by a large manufacturer as part of their pandemic adjuvant. And so what we can see here is that overall we're getting good immune responses. These are neutralizing antibodies, so very high titers, looks all great. So after 1 immunization is day 28, day 56, we clearly see that the vaccine, both vaccine candidates, are inducing high levels of neutralizing antibody titers. And then after challenge, there's hardly an increase, but then the control group is coming up, indicating that the animals have seen the virus. Next slide, please. But most importantly, when we now look at the viral infection in the upper respiratory tract, and here broken down now by the 2 vaccines and the control groups, you can see that only the vaccine that has the Th1 inducing adjuvant was really a balanced Th1, Th2 response. The adjuvant here makes a difference, and you can see 6 out of the 8 animals are essentially free in their upper respiratory tract, whereas only 2 animals have some viral shedding in their upper respiratory tract. Comparing this to the commercial adjuvant that is currently being used by many groups around the world, you can clearly see that the adjuvant here does not make a difference in this trial. Compared to the control group, there is statistically, except for this day here, there's no difference between these groups, whereas all of this here is statistically significant. So you can really see that the adjuvant makes a difference, and of course we all know that for subunit vaccines that the adjuvant is critical. In particular, though, for COVID-19, we think this is absolutely key in going forward in considering and this. There's a question here. These are ferrets now. This is in the ferret model. Next slide, please. And so here's our timelines. As I mentioned, we're in the middle of our safety and tox studies right now. We're going to do an NHP study here in October to end of December, and then in parallel going into phase 1 starting in October, and this will be done in Canada. Next slide, please. And that's my last slide. So thank you very much, and happy to answer any questions later on.
Thank you, Dr. Kurtz. May I ask Dr. John Lewis to start his presentation on vaccine-mediated enhanced disease? Dr. Lewis is the CEO of Entos Pharmaceuticals, a biotechnology company developing genetic medicines using the phosphogenix drug delivery system. Entos is developing phosphogenix DNA-based vaccines for COVID-19. Dr. Lewis.
Thank you so much, Anita, and thank you so much for the invitation to speak today. So I'm going to speak today quickly about an issue that's present in the minds of everybody developing vaccines, and this is the idea that we can enhance disease by trying to protect ourselves against it. So I just want to split it into two topics, really that are separate in their mechanism: vaccine-enhanced disease and antibody-dependent enhancement. Next slide. So vaccine-enhanced disease is more of an antigen presentation issue, and really exemplified by issues with the formalin inactivated respiratory syncytial virus vaccine developed in the 1960s. Inactivated RSV was administered to children, which made it particularly troubling. It was the inactivation protocol was performed with formalin, which unfortunately blocked its proper processing in the cytosol to produce proper antigen presentation. And what this unfortunately did was skew the response to a CD4 based response, Th2 skewed immune response, which then led to severely enhanced disease, hospitalization in many cases, and in some cases, in fact, mortality in these vaccinated infants. So at the time, the pulmonary histopathology was reported in the literature as a peribronchial monocytic infiltration with some excesses in eosinophils. We know a little bit more about that now. And these observations were further supported by reports of increased numbers of eosinophils and CD4, but not CD8 T cells in mice, and high levels of type 2 cytokines in mouse models. So really, this is an inappropriate T cell response in response to this inappropriate processing of the antigens. And as Volker mentioned, this has been addressed largely these days by the development of more advanced adjuvants and the development of genetic vaccines that produce the antigens in the cytosol. Next slide. So something that's present in the minds of many vaccine developers is this other issue, antibody-dependent enhancement, which is really important because it's directly tied to the mechanism of action of neutralization and clearance of the virus. And so typically this will, when you get vaccinated against a pathogen, you produce a variety of antibodies. Some of these are neutralizing, preventing uptake and infection. Some of these are non-neutralizing. Some of these may be actually enhancement, or enhancing, in that they will bind the virus, but then mediated through their FC receptors, be taken up in certain cells that express FC receptors like macrophages and certain other cells will express them as well, facilitating viral entry. And this can severely enhance infection and therefore disease. And this can also be mediated by a complement-mediated pathway. Next slide. And so I'm making a bold statement here, but certainly in our experience so far, and in our discussions with many, it seems like the in vitro assays don't do a great job of predicting vaccine enhancement, so the antibody dependent or otherwise. That said, though, I think we're in sort of this rapid development and knowledge gaining about SARS-CoV-2 specifically, and I think as specific mechanisms are identified for potential enhancement, we may be able to develop assays to address this. Next slide. And so we have some pretty good and growing preclinical evidence so far. And I'm going to just present some non-human primate data that suggests we have some optimism on this front. So in a couple of re-challenge studies, if you look at rhesus macaques that were re-challenged 28 days after their initial infection, and in particular focusing on the animals that had very low neutralizing titers and that had viral shedding. So no evidence of advanced or enhanced disease was seen in this experiment. Again, another cohort of 9 macaques that had neutralizing antibodies, antibody-mediated effector functions, and CD4 and CD8 T cells. All these were associated with protection upon re-challenge at 35 days without any evidence of ADE. On the vaccination side, these are just a couple of examples of many that have been published now. First, on the inactivated vaccine front, so macaques immunized with inactivated SARS-CoV-2 and alum had protection against a high inoculative of SARS-CoV-2, and in that study, no histopathological analysis had no evidence of ADE, and subsequently a larger study using raw plasmid DNA in 35 rhesus macaques with a variety of different constructs, including full-length spike and receptor binding domain, showed a really good correlation protection with neutralizing antibodies. And even though this is FCC receptor dependent effector functions, we didn't see any adverse outcomes with challenge. So this is the reason for optimism. Next slide. So a little bit about our platform. So it's a good segue. So we think that a DNA-based approach, I think, is compelling from the standpoint because we get proper good antigen presentation. Of course, with DNA, we have a lot more flexibility in the incorporation of antigens and antigen combinations, and it has a lot of compelling features for scale up and distribution. Obviously, DNA is very stable. If you look at what's out there, and I showed you an example of a sort of a raw plasmid DNA injection, but typically DNA is introduced either through electroporation or by using another virus like an adenovirus, as many of the clinical vaccines are using. Our platform is a sort of a hybrid between the two. So we use a lipid nanoparticle platform that's easily manufactured and scaled. But for the DNA entry, instead of using an ionic or cationic lipid for entry, we utilize a small virus-derived fusion protein dubbed the fusion-associated small transmembrane proteins, and these were discovered by a key team member, Roy Duncan at Dalhousie, a Canadian virologist. And basically, so we can formulate these lipid nanoparticles containing plasmid DNA, incorporating these fusion-associated small transmembrane proteins in the lipid shell, and these facilitate direct fusion of the DNA vaccine into the cytoplasm. Next slide. So starting in January this year, we started what we call the rapid prototyping process, thinking about generating both neutralizing antibody response, but also a potent and durable T cell response. So we looked at combinations of spike protein, N protein, and optimized epitopes. Next slide. And of course, we looked at in vitro assays, in this case, pseudotype virus assays, for looking at both antibody-dependent enhancement or neutralization. And as Volker mentioned, we're working with Vito Intervac to look at ferret challenge models, hamster challenge, and also we're looking at immunogenicity in non-human primates. Next slide. So just quickly some data. So we've encapsulated now a variety of different plasmid constructs in what we're dubbing a COVIDgenics vaccine using this fusion-associated small transmembrane platform. We're able to elicit with so this is a 2-dose regimen given on day 1 and 14, 25 microgram dose where we think the MTD is about 2 milligrams. We're getting a very strong T cell-based response. Next slide. And with the ZEST vaccine, and I'm sure we'll talk about assays later, we got potent neutralizing antibody response. And we looked at a panel of convalescent patients, about 20 patients. We got with the full-length S spike vaccine, we got equivalent neutralizing antibody at this low dose to convalescent patients. Next slide. One thing we're really interested in as well is the development of a pan-coronavirus T cell vaccine. And these are based on, in this particular case, a full-length N construct. It's early days with this, but at the same low dose of 25 micrograms, we're getting a potent CD8 response we can see in lymph nodes and spleen. Next slide. So I'll finish off there. I think we have a long way to go as far as developing in vitro assays, and it may not happen. I think it's critical to establish correlates of protection. We've seen in our studies as well that overall antibody anti-spike response, for instance, is well correlated with neutralizing response. I think this, as we move on, we're going to be able to do mechanistic studies of either correlates of response or enhancement in animal models. And I think we have a note of optimism that the preclinical evidence to date suggests that our modern vaccine approaches with modern adjuvants and genetic approaches have minimized the likelihood of enhanced disease. We'll hope that holds true because we're heading into the clinic to phase 3 studies. Thank you very much. And last slide, sorry. So I represent a fantastic team of Canadians and throughout North America, and we're looking forward to starting our phase 1, phase 2 studies in Canada with the Canadian Centre for Vaccinology in August or September this year. Thank you very much.
Thank you, Dr. Lewis. Next, we have Dr. Sarah Gilbert from Oxford's Jenner Institute. And Dr. Sarah Gilbert is the project lead for the ChAdOx1 adenovirus vector vaccine that I'm sure many of you have heard about. Thank you, Dr. Gilbert. All yours now.
Thank you. So today I'm going to talk about immunogenicity, how to measure immunogenicity in the vaccine trials that we're all running. Just a little bit of background on the technology that we're using. It's the simian adenovirus vector vaccine ChAdOx1, replication deficient. So, and it expresses full-length spike protein. It doesn't replicate after vaccination because the E1 gene has been removed. That needs to be supplied in trans during manufacturing in the cell line that's used for manufacturing. The E3 gene has been removed as well, which gives more space for adding in a transgene. So we're using a simian adenovirus because this avoids issues with pre-existing immunity to human adenoviruses, and the pre-existing immunity to this viral vector is very low in Europeans, and we will be studying that more across the world as we roll out the programs that we're currently running. The vaccine antigen is in the DNA of the adenovirus. It's not a structural part of the virion, so this is a true platform technology. And the vaccine antigen, the full-length spike protein in this case, is not expressed until after the vaccine has been administered and entered a cell, and then the spike protein will be expressed inside that cell to be processed in the same way it would be if the cell was infected by live coronavirus. That results in an immune response which induces strong B and T cell responses after single vaccination, and that's what we then want to measure in our immunogenicity experiments. So we have considerable experience of using this platform technology before using it in SARS-CoV-2 vaccine development. Prior to April of this year, we had conducted 12 phase 1 studies with 330 subjects vaccinated with different antigens in these vaccines. But simian adenoviruses in general, as opposed to just ChAdOx1, had been used in clinical trials of over 6,000 subjects of all ages, from infants in malaria vaccine trials to older adults in flu vaccine trials. And we've seen a very consistent safety profile and strong immunogenicity after one dose in these trials. More relevant to SARS-CoV-2 vaccine development, we'd also been working on a vaccine against another coronavirus, the MERS coronavirus, and as is now published, and you can see the references on the bottom of the slide, single dose of ChAdOx1 MERS was protective in non-human primates, and it has completed a phase 1 study in the UK, which has been published, and there's an ongoing phase 1 study in Saudi Arabia. Next slide, please. So to illustrate the assays that we're using to measure immunogenicity, I'm going to present you with some figures that have already been published in a preprint. Again, the link to this is below. This should shortly be accepted as a full paper. And in this study, we looked at the administration of 1 or 2 doses of ChAdOx1 nCoV-19 in pigs. This is purely an immunogenicity study. There was no attempt to challenge the pigs to test vaccine efficacy or look for enhanced disease. We're just looking at immunogenicity here. But a wide range of assays were used, and that's why I've chosen this to illustrate the ways we can measure immunogenicity in vaccine trials. So starting from the assay on the left, ELISA, workhorse assay in many labs, where we're measuring antibodies that bind to the full-length spike protein. And what we have here is we can do this on multiple days on the day of vaccination and then multiple days post-vaccination, and we can follow the development of the antibody response in animals that received 1, which are the open circles, or 2, which are the closed circles doses of the vaccine. And we can see a nice induction of immune responses after the first vaccination, and in the animals who receive a second, we can see boosting of that response. So there are no units given on the y-axis here. And that's deliberate. That's because depending on the way you set up the ELISA in your own lab, you will get very different results. You may get a certain amount of variability between assays run on different days. And although this type of assay is fine without use of standards and calibrators in a preclinical study, when we move into clinical development, if we're measuring binding antibodies to the spike protein, we need to be much more careful about validating and standardizing the assay. So all of the parameters that are in use in the experiment need to be tested, and the upper and lower bounds for various parameters need to be determined. So that means determining the right amount of protein to coat the plate with. If we're doing a direct ELISA, as this is, what will the secondary antibody be? What will the incubation times be? All the steps of the assays, and testing those and testing how much you can vary from those parameters and still achieve the same results. And in order to standardize, we need to include the same amount of the same control serum sample, or possibly a monoclonal antibody, but it's better if it's a serum sample, a polyclonal serum, to be included in the assay every time. And then all of the results are normalized based on the readout from the standard. And that enables us to get results which are comparable across days in the same lab, across operators in the same lab, and then eventually between labs, which may be in different countries. We should also be looking to include calibrators. So having standardized or normalized on the standard, we would then want to see that the calibrators, which are usually low, medium, and high responding serum samples, will give us a very small range of variation between different assays after the standardization has been applied. So in this experiment, it's a first look-see to see how the antibody response is developing and does it change with a second vaccination. But to apply this for clinical trials, we need to do a lot more work on the assay to get to a standardized and validated assay. But we're also very interested in neutralizing antibodies when we're thinking about SARS coronavirus, because this is everybody's best guess at a correlative protection. That hasn't actually been determined formally as a correlative protection, but this is the type of immune response that everybody believes will be most likely to correlate with protective efficacy. And there are many different ways of doing neutralizing antibodies. One of the ones that is done is to look for complete absence of cytopathic effect in wells of vero cells, which have been incubated with a mixture of diluted serum and virus. And so that's looking for 100% absence of cytopathic effect. It's a very stringent assay. And so the serum titer, or the serum dilution that will achieve that, tends to be much lower than what's represented here, which is an ND50 assay. So here we are looking at the serum dilution that achieves 50% reduction in the number of plaques in setup in the well. And the normal range would be to have about 100 plaques caused by SARS-CoV-2 virus in the wells where there was no serum applied, and you're looking for a reduction down to 50 plaques in the wells where diluted serum is applied. And this will be done over a 6-point or an 8-point dilution range, and then the values will be used to calculate the ND50 or the IC50 titer. So here again, you can see that there is an immune response being generated, and that it's stronger after 2 doses than it is after 1 dose. And there is some variation between the animals, and that will be expected because these are outbred animals. This is using live SARS-CoV-2 virus, so this needs to be done in the containment level 3 lab, which means that there's a limit to the number of assays that can be run. However, it is often the case that if you look for correlation of other serological readouts with a live neutralization assay, you will find that they correlate very well. So having demonstrated correlation, it's possible in the future to use a different serological readout that doesn't require use of a containment level 3 laboratory for every single serum sample. So next slide, please. So on the right here, we have an example of that, a pseudotype neutralization assay, where a virus other than SARS-CoV-2, but carrying the spike protein on its surface, is used in the same way. And that doesn't require high containment, and if done well, can be correlated with the neutralizing antibody assays. On the left is a microneutralization assay, and what that means is you are looking in a very rapid assay for the reduction in the number of single infected cells after a 20 to 24-hour incubation. So instead of looking for reduction in plaques, you're looking for reduction in infected cells, which is essentially the same type of readout that can be done much faster and is more likely to be able to be automated, both in setting it up and reading the results. And the next, please. We also need to look at T-cell responses, and the assay that is most commonly used in clinical trials is the interferon gamma alispot assay, looking at cells which release interferon gamma in response to stimulation with peptides spanning the whole of the antigen that's being included in the vaccine. And this is something that can be validated, standardized, and rolled out quite easily. And the next slide is looking at intracellular cytokine staining, which can give you a lot more information about the release of different cytokines, but is much more difficult to standardize. And next, please. So in order to run these assays, we have certain requirements. We need protein, we need peptides. These should be the same across different labs. We need standards and controls. Negative and positive controls are standard in the calibrators to compare the assay. And we need core labs that are able to run these assays. So when we get into large-scale clinical trials, we eventually reach a situation where we can truly compare like with like for assays measuring binding antibodies, neutralizing antibodies, and T-cell responses. And I will stop there. Thank you.
Thank you, Professor Gilbert. Our final speaker in this session on immunity and vaccine-mediated enhanced disease is Dr. Jeffrey Knowles from the Oregon Health and Science University in the US. He focuses on T cells and cell trafficking and overall function of the immune system. And he's going to talk to us today about long-term immune memory. So, Dr. Knowles.
Thank you, Anita, and all the other organizers for putting together this lovely discussion. Next slide, please. So, my lab doesn't really focus on vaccine development per se, but really has strong interests in understanding the basic mechanisms of T-cell activation with a very strong interest in the development of long-term immunological memory. And so the graph I'm just starting off with here today you can find basically any textbook where this illustrates the activation of the adaptive immune response, in this case, the activation of cytotoxic CD8 T cells, where once those T cells are primed, they undergo this rapid proliferative expansion phase, differentiation into effector cells, followed by a contraction phase and the development of long-term memory. And if we want to start thinking about what are the features of immune cells that are the fundamentals of memory T cells, they are listed there on the right. And so, of course, one aspect is that there's more of these cells. They're going to have immediate effector functions, which means they can produce cytokines and undergo cytotoxicity within seconds of re-encountering their antigen. These cells have different tissue infiltrating capacities. Importantly, they are very durable and long-lived. And if together, these cells are able to then provide robust protection. Next slide, please. So in my lab, one of our favorite models to study is the use of a virus called vaccinia virus, which is a large DNA virus of the pox virus family. One reason we are interested in this is, of course, the smallpox vaccine. And so arguably you could say this is the most successful vaccine in the history of mankind. So understanding the mechanisms that cause the activation of the adaptive immune system downstream of this viral infection, we can start to understand what are the hurdles that are going to be necessary to overcome to provide robust protection of immunity. Importantly, this is a virus that's very easy to work with, and so you can make recombinant versions of this virus that are able to express either model antigens or fluorescent proteins if you're interested in basic mechanisms of the laboratory, but also foreign antigens as a vaccine candidate. And the example I'm showing here at the bottom is the activation of the CD8 T cell response in response to a viral skin infection with vaccinia virus. And so if you just take naive black 6 mice and infect them on the skin with vaccinia, look 7 days later, you can see in the draining lymph nodes, you have robust responses against the immunodominant epitope. In this case, it's the B8R 2027 peptide. You get degeneration of circulating memory, as you can see in the spleen, and the cells can also traffic into the skin, shown there on the right. Next question, or next slide, please. To demonstrate how effective the smallpox vaccine was, this is some data that a colleague of mine, Mark Slifka, out at the Oregon Primate Center published quite a few years ago now. And it looked at the identification of immune cells that were specific to pox virus in individuals that were vaccinated up to 70 or 80 years earlier. Now, it's important to know that in 1980, of course, the eradication of smallpox was declared by the World Health Organization. In the United States, the smallpox vaccine was stopped giving something around in the early 70s. And so the assumption here is that people aren't getting re-exposed to pox viruses. And so the generation of these immune cells occurred strictly by the vaccination. And if you can see on the left graph there, the development of IgG antibodies against pox viruses is incredibly stable in almost every individual. And the cellular immune response shown there in the right two graphs for the CD4 and CD8 T cells are generally stable as well. So we know that this is an effective vaccine. It prevented disease, and there's correlates with immune protection, as we can see with T cell responses and humoral responses. Next slide, please. So to get activation of certain types of immune cells, there needs to be, of course, antigen presentation. And so the activation of your cytotoxic CD8 T cell populations occurs primarily through MHC class 1 presentation, which delivers antigenic peptides into the ER through transportation of the TAP complexes, and presentation on MHC class 1 and of course, class 2 peptides are generally thought to occur by the uptake of exogenous antigens. Delivery of those peptides from the endosome into an antigen processing compartment, where they displace the clip peptide and then can be presented to CD4 T cells. And the activation of CD4 T cells is, of course, important, because if you want to get high affinity class-switch antibody, there needs to be the delivery of CD4 T cell help to activated B cells in order for them to differentiate into long-lived plasma cells that go onto the bone marrow. Next slide, please. So a few years ago, we were interested in trying to develop reagents that would activate CD4 T cells in vivo using the vaccinia system. And what we found is that if you deliver short peptides expressed by vaccinia that should be presented on class 2, that system alone isn't sufficient to get robust T cell activation. And that's shown on the second histograms there, where if we infect mice with vaccinia expressing just a peptide from LCMV called GP61, using proliferation of CD4 T cells as a readout, there's no activation of these T cells in vivo. So we went to the literature and applied a couple strategies that are predicted to enhance MHC class 2 presentation. And that includes flanking peptide sequences with either LAMP1 to target the peptide to the lysosome, or by conjugating it to the MHC class 2 invariant chain, which is predicted then to pull that peptide into the antigen processing compartment. And as you can see, both of those strategies significantly enhance CD4 T cell activation in vivo, and in our hands, at least, the conjugation to the class 2 invariant chain seems to be the big winner. Next slide, please. So by what those data basically told us is that we could maybe start applying these strategies to, of course, the SARS protein. And so there's just a simple schematic here of the SARS spike protein with its S1 and S2 domains. Next slide, please. And so we went and made some viruses that expressed either the full-length SARS coronavirus spike protein or SARS coronavirus spike protein that was conjugated to the invariant chain. When we infected cells in vitro with these viruses, we got this kind of surprising or unexpected result where the virus expressing the whole-length spike protein, we could clearly see expression in vitro, but we would not see the expression of the virus that was attached to the invariant chain. But you'll see in a couple of slides this maybe for other reasons. Next slide, please. So a simple experiment we performed is we infected mice with either a vaccinia virus control, a vaccinia virus expressing the coronavirus spike protein, or conjugated invariant chain, naive black 6 mice, and then we wanted to quantify the immune response that was mounted against those viruses. Next slide, please. Using some peptide pools showing here, and I want to focus down on the bottom 2 graphs, looking at this is the CD8 T cell response mounted against either what's predicted dominant epitopes of the full-length S protein or the S1 domain. And you can see that both of our viruses, both the one that is expressing full-length S as well as S conjugated to invariant chain, are both eliciting very strong CD8 T cell responses as shown by the production of interferon gamma. Next slide, please. And we are also able to monitor CD4 T cell responses. And so what basically these data show is that even though the coronavirus spike protein conjugated to the invariant chain doesn't seem to be stably expressed, it is clearly stimulating T cell responses in vivo. And so there is still this prediction that there could be enhanced CD4 mediated antibody production using this system. Next slide, please. And so this is, of course, early work for us, but we basically have shown that we have generated recombinant vaccinia viruses that express the SARS coronavirus 2 spike protein. We show that the conjugation of the full-length spike protein to the invariant chain may actually lead to more stabilization or degradation of the protein, and that we're currently investigating that. Immunization with the vectors, as we can show, activates both CD8 and CD4 T cells, and the dominant response may be against actually the S1 domain and not the S2 domain. And finally, the development of long-term immunological memory and neutralizing antibodies, of course, are currently ongoing in my lab. And thank you to everybody, and I'm all finished.
Thank you very much, Dr. Knowles. So that concludes the 4 presentations. We're going to be starting the panel discussion now. I am going to start off the panel discussion, and then we will get to the lots of questions that we have in the query section of this presentation. Let me start by, so the $1 trillion question is, how do we get durable SARS to CoV immunity with an acceptable safety profile? So I'm going to pose the first question to Dr. Gertz. Can you speak more broadly about what we are learning from animal models about vaccine efficacy? Do you think we will be able to prevent infection in the upper airways, which then means that it's a transmission blocking vaccine as well? Or can we only hope to get prevention of severe disease against severe disease? So could you address that question, please?
So in our preclinical, can you hear me there?
Yeah.
Okay. In our preclinical models, it looks like it's going to be very, very difficult to prevent complete viral replication in the upper respiratory tract. As I pointed out on this one slide there in the ferret model, one of the vaccine candidates that we have here seems to be highly effective in reducing viral replication in the upper respiratory tract in most animals compared to the other one. So the type of immune response is critical, and I believe that you have to have a good combination of neutralizing antibodies as well as T cell immunity. I think neutralizing antibodies alone will not prevent viral replication in the upper respiratory tract. And so what can animal models really offer? So I think that's one perspective. I think, your question also suggests, or, like highlights that immunity may be short-lived for this one. So again, our animal models are now being used to look at duration of immunity. And, unfortunately, not all the reagents are available for ferrets and hamsters. But I think it's the combination of the type of immune response and neutralizing antibodies that make the difference.
Thank you. And Dr. Lewis, quickly to you, which animal models do you think are going to be turn out to be most useful in understanding safety issues, vaccine-mediated enhancement, for example?
Oh, that's a good question, and something we've been sort of discussing a lot. Initially, even in my slide is reflected, we were initially going with the ferret models. We're working closely with Volker and the group at Vito. It looks like the hamster model seems to get a more fulsome disease with maybe better correlative pathology. Most importantly, also, they seem to lose weight after infection, which is a great stable readout. So I think, as far as infection and challenge, and comparing with non-human primates, the challenge models in non-human primates, we don't have any direct experience with them yet, but they seem to be pretty variable with variable pathologic response. So in our group, we're thinking about using hamster as a challenge model, given its more fulsome pathology and the weight loss, and then and as far as safety goes, we're doing all our safety studies in non-human primates. We feel this is the closest to humans.
Great, thank you. So I have so far 15 questions in the Q&A. What I'll try and do is pick 16 now. I'll pick ones that cover the whole session. So let me, I may not be able to do all of them, but I am told by the organizers that the panelists will have access to these questions and can get back to people whose questions did not get answered. So this is a question from Marion Koopmans. She's asking, other than specific neutralizing antibodies, is there a common understanding or consensus on what possible other immune correlates to study, including the potential for adverse effects? So this is, again, a question for the first two speakers.
So again, I think the role of T cells is going to be critical. So T cell immunity, in my mind, is important for protection.
Dr. Knowles, do you also want to address this? Because I think that there is a lot of chatter about T cells.
So I would say that, of course, in the media and stuff, everybody wants to throw around that, of course, the development of neutralizing antibodies is the gold standard for vaccine efficacy. And we've used that as a correlate for almost every type of vaccine. And, the T cells generally get ignored. And we know in many of our models, even in wild type mice, that if you get rid of memory T cells, even in the background of neutralizing antibodies, efficacy of immune protection gets lost. And so, you can use neutralizing antibodies as a readout for potentially the development of strong T cell responses, but knowing whether either a cytotoxic T cell response or a CD4 helper T cell response is certainly going to be almost certainly necessary. And that was what we kind of talked about in my presentation, where how can we try and deliver antigens into one compartment or the other to boost those responses. Certainly studies from Stanley Perlman's group at Iowa can demonstrate that CD8 T cells can be sufficient to provide protection. Whether they're necessary is something I don't know that we necessarily understand yet, especially against this novel coronavirus.
Thank you. Dr. Gilbert, this question is for you from Manuel Barrel Neto. What are the planned correlates for protection for testing the proposed vaccine preparations?
So during vaccine development, we should be looking at as many different immunoassays as possible. We obviously need a readout on neutralizing antibodies, but we're not going to be able to do neutralizing antibody assays on tens of thousands of individuals at multiple time points. So we need to be able to find something that correlates with that. And normally what you see, if you look at other vaccines that have been in development, it's possible to get a well-standardized ELISA or another serology assay that doesn't require high containment, that correlates well with the neutralizing antibody response. And that's most likely to be used as the eventual correlate of protection for that vaccine. But vaccine correlates of protection are vaccine specific. And if we only measure this serology response, we're ignoring the T-cell response. So for any particular type of vaccine, whether it's a viral vectored vaccine, a DNA vaccine, a protein subunit vaccine with a particular adjuvant, there will be a fairly standard relationship between the CD4, the CD8 T-cell response, the ELISA titers, the binding antibodies that is, and the neutralizing antibodies. And that needs to be understood for each type of vaccine. Once we understand it, the eventual assay for correlative protection is likely to be something that's simple, easy to standardize, and easy to perform on a very large scale, and it will probably be a measure of binding antibodies, possibly binding antibodies specific for the RBD domain in S1, but or it could also be for the whole spike protein.
Thank you. So for the benefit of our participants, a point that I would like to emphasize is it's really important to understand that this is going to depend on the type of vaccine, what platform you're using, and it's going to vary by, so we probably will not be able to have one correlate of protection. It will depend on the platform. And for a good understanding of this, the supplementary material in the NGM paper on Moderna is a good one to read through and compare to what the Oxford folks have put out. There's another question for you, Dr. Gilbert. This is from Manoj Das. You partly addressed this, but he has a second part as well. So for detecting the protective antibody titer and vaccine efficacy, which antibodies are being considered? The antibody response in several infected individuals, so infected individuals are low. What relevance does this have for vaccine efficacy testing?
So the vaccine correlate of protection needs to be determined in phase 3 trials of vaccine efficacy. We can't assume vaccine efficacy by reaching a particular level of antibody response, of any type of antibody response. We need to actually determine efficacy in trials where we have a control vaccine and the test vaccine, and follow subjects and count the cases. And then from that, once we have determined vaccine efficacy, then we work back to the correlates of protection for that particular vaccine. And then we can start to think about whether that correlate may apply to other vaccines in development if they're of the same type. And it will give us some indication of the correlates for other vaccines as well. But as we've been talking about, if there's a strong T-cell response, it may be that the neutralizing antibody titer required may be somewhat less than a different type of vaccine that gives neutralizing antibodies in the absence of a strong T-cell response.
Yeah, thank you. So I think that what the person is also trying to get at is that they're a little bit worried that in convalescent humans recovering from disease, that some people have low antibody levels, and what does that mean that we might have to do better than natural disease for protection?
So the mechanism of induction of these immune responses is not necessarily exactly the same in people being infected with SARS-CoV-2 and people being vaccinated. And if you look at vaccine trials using viral vectored vaccines and DNA vaccines and other types of vaccines with other antigens where follow-up has extended for at least a year, we see good maintenance of antibody responses. So in particular, I can talk about the ChAdOx1 MERS trial, where we followed subjects up to a year and saw good maintenance of both T-cell and antibody responses a year after a single vaccination. So that's not what we're seeing in people infected with SARS-CoV-2. It doesn't mean that we won't see good maintenance of antibody responses after vaccination. And we need to determine that, but we shouldn't be too despondent about the fact that antibody responses seem to be declining quite quickly after infection. It's not necessarily going to be the case with vaccination.
Great, thank you. So this question doesn't, they did not tell us who they are, but the question is for Dr. Lewis. Says in 2007, an adenovirus-based anti-HIV vaccine STEP trial sponsored by Merck appeared to increase the rate of HIV infection in individuals with prior immunity against the adenovector used in the vaccine. Will the new platforms projected for COVID vaccine be tested for such adverse effects? This may concern the adenovirus-based and possibly other DNA and RNA-based formulations.
Sure, yeah. So I'm not intimately familiar with that particular study, but I think every vaccine developer is aware of the enhancement studies and is actively integrating them into their testing plans. And I'm sure that's no different for the adenovirus or the other RNA or DNA platforms.
Great. And then there is another question from Nesreen Ozeran. Any one of you can take this. Do you think this G614G mutation will affect the spike protein immunogenicity?
It's been shown that it doesn't. Antibodies that neutralize the original sequence neutralize 614G bearing spike proteins equally well.
Okay. This is interesting. Also anonymous. Pfizer and Moderna vaccines showed reasonable level of mild to moderate adverse events in phase 1. Is this a necessary evil to generate high titer neutralizing antibodies, or do we expect that the more traditional approaches, such as the adjuvanted protein vaccines, to generate a more suitable safety profile? I guess I'll have Dr. Lewis address that.
Well, so this is the issue with developing vaccines. The very immune response we're trying to elicit can sometimes have systemic effects. So I think this is potentially an indication of efficacy, although there'll be maybe specific adverse events that are specific to certain delivery systems and platforms.
Great. Okay, and then one final one that I think we have time for is for Dr. Knowles. What markers of T cell memory were used in the preclinical studies in the studies shown? Are some evaluation of T cell memory available from the clinical studies already initiated?
So in some of the limited stuff I've seen from people like Shane Crotty and Alejandro Cedre and La Jolla, can certainly demonstrate using ex vivo peptide stimulations that there are both CD4 and CD8 memory generated in patients recovered from COVID-19. They use a variety of markers in addition to cytokine production to demonstrate that biology. The other somewhat interesting thing that they showed in that, it's a Cell paper if anyone wants to look it up, is that many uninfected people seem to have CD4 T cell responses that are presumably generated from other coronaviruses already present. And so this is somewhat of an interesting finding that perhaps there's cross-reactivity in the CD4 compartment for SARS coronaviruses and also your more widespread coronaviruses that give you seasonal cold, for example. But it didn't seem like that was necessarily the same thing for the CD8 T cell response. And that actually might be somewhat interesting which also may suggest that CD8 immunity might be important for the prevention of reinfection of this type of virus.
So that's helpful. One of the issues with Dr. Gilbert covered this a little bit. I mean, assay standardization is such a critical element, and we are so early in all of this, right? We're trying to build the plane as we are flying, and so T cell assay variability across the different, we don't know how to measure T cell response across and compare across constructs.
Sorry, I think the other important point here is that for people, we have to use blood as basically a marker of immunity. Whether the same immune response is present in non-lymphoid tissues is almost impossible to predict just based on that analysis.
Exactly. So there's a lot of unknowns here right now. And only the phase 3 trials tell us.
You're very difficult to hear at the moment.
Sorry, can you hear me now?
Yes.
Yeah, okay. I'm saying only there's so much that's unknown right now about this. And so only getting into the phase 3 trials and understanding how the how the different products perform over time, will many of these questions be elucidated. Let me just pose this one last question before we move into the rest, and this is to any one of you who wants to answer it. What are the remaining gaps, and what can GLOPIDAR members do to fill them in our sort of preclinical space? Can you hear me?
I can take a stab at that one. I think this has come up multiple times in the conversation. I mean, I think we need standardized assays to determine correlates of protection, both for neutralizing antibodies and for cell-based responses. And I think, as we move forward through clinical trials, these assays will be essential.
How about in the clinical space, Sarah?
Oh, I was just going to say a bit more about preclinical space. So one thing that we haven't really, where animal models can also help, is in understanding transmission. So what we're seeing is when we have a macaque study where macaques are being exposed to a massive dose of virus, which is being delivered by multiple routes, and then you see virus coming out of the nose. Actually, that still happens if you put dead virus into the nose. You can measure the same amount of viral shedding if you put irradiated virus into the nose of a macaque and follow the shedding over several days. So if we're doing vaccine studies which are designed for safety, and then we're seeing loss of viral RNA from the nose, that's not saying that that virus doesn't present transmission. So what we do need is a different setup of the animal model to look at transmission. Having established safety, having established some degree of efficacy against disease in the animal that's vaccinated, we then need other animal models to assess will those vaccines prevent transmission if the animals who've been vaccinated are then challenged to co-house with naive animals.
Okay, great. Any other quick response? We're out of time actually. So I mean, just if I'll take a 1 minute from Dr. Kurtz or Dr. Knowles.
Nothing really to add. I think determining the right, the correlates to determine the right protective type of immunity is critical. And so again, standardization of T-cell assays will be critical.
Okay, with that, let me move into the second session of this, which is manufacturing, implementation, and access, subject very close to my heart. I will ask speaker here is Dr. Yadav, who will talk to us about he'll talk to us about manufacturing. He's from he's a globally recognized scholar in the area of healthcare supply chains, has a PhD in management science, and currently is a senior fellow at the Center for Global Development, a professor at INSEAD, and a lecturer at Harvard Medical School. So would love to hear your thoughts on manufacturing, Dr. Yadav. And this is a super fast session, 5 minutes per speaker. So we're challenged for time here. Thank you.
Thanks, Anita. I'll try to be as precise as possible. Next slide, please. So I think similar to the state of affairs in global collaboration and cooperation around science, colleagues who work on manufacturing science, so in a company, people who lead the technical operations or tech ops, or those in philanthropy or at CEPI who work on CMC, have been working around the clock to understand what is the state of manufacturing capacity, where is it located, how much of it is available, how much of it will need to be reconfigured. So lots of important, relevant, but challenging questions. And what I'm hoping to do in the next 3 minutes is give you a quick overview of what our current understanding is. And when I say our, I imply that me as an academic who is largely observing much of this from the sidelines, this is a viewpoint that I'm sharing with you. So if we think of manufacturing capacity, I think it's worth thinking about 3 things which are very connected, but still somewhat separate. So the first is manufacturing capacity for drug substance. The second is manufacturing capacity for fill and finish operation. The third is availability of input materials, including adjuvants, stabilizers, vials. And then the last is distribution capacity once we have a vaccine manufactured, and each of them is equally important. So let's start with drug substance manufacturing. We know this is highly platform dependent, and the steps required change depending upon which vaccine platform are we looking at, and the degree of interchangeability and fungibility of manufacturing capacity from one platform to the other is very limited for certain kinds of platforms and is reasonably high for other platforms. So what we've seen in the last 3 months is multiple deals, some of which have been executed bilaterally between a vaccine developer and contract manufacturers. Others which have been between multilateral organizations such as CEPI or from country governments, in primarily the ones in high-income countries, with specific vaccine developers to enable them to make at-risk investments in capacity. The manufacturers or the vaccine developers have in turn contracted manufacturing capacity to CDMOs or contract manufacturers. And it remains unclear as to what extent can such capacity be quickly reconfigured in case that vaccine candidate does not meet our TPP or the required efficacy and safety standards, and we want to then use that manufacturing capacity for an alternative candidate, because we don't necessarily have the luxury to invest in capacity at full scale. And by full scale, I'm implying, whatever is the cohort size of 5 billion, 4.5 billion, some number like that. So we do want the ability to use the same assets for the candidates which are more successful. So in that sense, it requires a portfolio approach to thinking about manufacturing capacity. And when we think about drug substance manufacturing, the other part that stands out is that for some of the newer vaccine platforms, the manufacturing capacity that either currently exists or the new production suits that are being put up based on the contracts that contract manufacturers have received, they are largely concentrated in a few countries, which are largely high-income countries. Then when we look at fill and finish manufacturing, there we see a higher degree of fungibility or interchangeability across candidates. Again, the capacity depends upon the type of packaging we have for a given candidate, which means if it's glass vial, is it BFS, is it something else? And also to some extent on the number of doses to be packaged in a vial. The fill and finish capacity is spread globally. I think it exists in a much wider global footprint than what we have for drug substance manufacturing. But the countries that stand out with large capacities in fill and finish are India, US, China, and Europe. And the countries where fill and finish capacity does not exist or is in very small dose equivalents are in Latin America, Africa, and Australia. Once again, I think if we look at fill and finish capacity in the aggregate, which means we look at what our projected global demand is and what is the aggregate availability of fill and finish, and this would include things such as sterile and parenteral manufacturing plants, which are not currently dedicated to vaccines, but are reconfigurable to make to do fill and finish for vaccines. If we include all of that, we have enough, most likely in the aggregate. But again, the important point here is that the aggregate can only fulfill global demand if we take the production out of the aggregate and then allocate it. But if we think about individual geographical boundaries, then clearly we may not have sufficient fill and finish capacity. The other part to keep in mind is that while a lot of public dollars have gone into creating capacity through contracting, we still are not sure if auxiliary inputs, especially vials, and in some cases adjuvants, do we have sufficient capacity for that? And are companies investing in building capacity at risk without necessarily having commitments from the vaccine developer and the contract manufacturer, or from public agencies. And so that's an area where I think we probably need to do some more work. And then the last thing is a question mark, which is if we have fill and finish manufacturing capacity and we resolve our drug substance manufacturing constraints, then we would still be faced with a big challenge in terms of cold chain storage and distribution capacity, which is something that shouldn't be necessarily decoupled from our questions about vaccine manufacturing capacity. So I have 4 things that I want to conclude with. One is it's important to look at supply network coordination at the portfolio level, which means the ability to change manufacturing sites, production suites from one vaccine candidate to another as we learn more about phase 3 study results, as we learn about as new information becomes available. That ability to change will be extremely important to match our limited capacity with a wide and growing portfolio of vaccine candidates. Then I think we need better purchasing coordination and equitable allocation rules so that we can resolve the regional imbalances that exist in fill and finish manufacturing. We need greater demand side predictability so that those vaccine developers who have not received a direct grant from CEPI or from any of the bilateral country national government mechanisms do see what is the demand at the end of this, and the 3 things that we ought to focus on there is better demand forecasting, using some kind of advanced purchase commitments, and certainly pooled purchasing, because when the demand is fragmented across multiple smaller countries, it becomes harder for a vaccine developer to convince their investors that, yes, this is worth investing in manufacturing capacity already. And then the use of development finance, which is whether it is IFC or UK CDC or other such development finance institutions, can they provide capital for scaling up manufacturing? I'll pause there and yeah, any questions, comments, or even insights that colleagues want to share, welcome that. Thank you. Back to you, Nita.
Thank you, Dr. Yadav. I hope you can stay for the whole session because we do have time for the questions at the end. We're bunching up the 2 presentations and then we'll come back to you. That's very fascinating. And there's so many issues to talk about here. So, okay, thank you very much. So we will go to Dr. James Love, who is going to load his presentation. There it is. He's the director of Knowledge Ecology International, and his training is in economics and finance. And he is his focus is on financing of research and development, intellectual property rights, prices for and access to new drugs, vaccines, and other medical technologies. Dr. Love. You're muted still.
Oh, I'm sorry. I'm not a doctor, so I just wanted to clear that up. I'm going to share some slides on my screen, and put this into a present mode. Can you?
We can see you.
We can see the slides right now. Okay.
Yes, we can.
Very good. Okay, I'm going to. Our organization it was started in 2006. We really incorporated work that was from another organization. We spun off from and kind of redefined the nature of the mission. We focus really on the way knowledge is managed. We have offices in Washington, D.C. and Geneva, Switzerland. Our work on access to medical technology is funded by the Pearls Foundation, the Open Society Foundation, Kaiser, and Unitaid. I want to start with very briefly with this issue, this issue of what constitutes a public good. And I mention this because there have been a number of interventions by governments and by NGOs, such as Doctors Without Borders or the South Center, MSF, leaders of certain heads of state, where they've talked about the idea that a vaccine should be a global public good. It was actually brought up in the WHO negotiations in the resolution they had at the World Health Assembly on COVID-19 about whether or not the term in these UN discussions, if vaccines should be described as a global public good. And in those cases, the US and some other countries objected because they said that term was not well defined. And there was a distinction that was drawn between the know-how of, or I should say, of vaccination itself, which was perceived to have a global public characteristic, goods public characteristic. Well, the idea being that if everyone's vaccinated, or the more people are vaccinated, the greater the benefit for the world. But there was some argument about whether the vaccines themselves as products would be considered a global public good. And that came down to really to an old discussion by an economist who won a Nobel Prize, Paul Samuelson, for a paper he wrote a very long time ago in 1954, where he defined what he described as a pure public expenditure as something that was non-rival in consumption and non-excludable. And he used examples, people have used examples as things such as lighthouses, national defense, outdoor circuses, a very limited set of goods. But the idea, if you have something where the number of people that consume it doesn't affect the supply or it doesn't create shortages, and you can't exclude people from having it, then it would have this characteristic. And it was really designed. And a lot of you aren't economists. My background's in economics, so this becomes a big, huge problem in the policy wonk field of people that do economics. But they feel kind of awkward about this term. Now, most ordinary people that don't read the old economic literature, they think global public goods are just things that governments do, and I think that's the right definition. I just wanted to point out that there's a fair amount of misunderstanding of Samuelson's earlier definition. He was describing what he considered a particular extreme case, but not the only cases that something could be defined as a global public good. And he's made that very clear in his writings. And I've written about this recently, and I have a citation at the bottom of this slide for a recent article on this. Now, I want to talk briefly about the World Health Organization's. They have a COVID-19 technology access pool that they've tried to create. They've kind of launched it basically in May. It was designed to provide the global pooling of rights and inventions, copyrights, designs, data, cell lines, and know-how, which is a very broad definition of intellectual property or things related to intellectual property. And they wanted to apply to all types of COVID-19 technologies, not just vaccines, but everything from protective equipment, diagnostic tests, and therapeutics as well. It is, as you know, and this is your own group is one of many international initiatives right now that related to both the research and access to COVID-19 technologies. I list a few of them on this slide, and there's more, certainly, including your group and others as well. And then there's a whole series of national initiatives, or sometimes described as bilateral initiatives on this area. And I think this is an interesting part of the whole effort on how you finance and how you manage the access. We've had discussions with countries trying to engage them about what their policies might be on access or what kind of collaboration they might be with the WHO. I think that most of the action has not been at the WHO on the IP front. There's been a discussion at the WHO that's primarily about who gets access to products without a lot of detail about how rights and patents or rights and data or rights and know-how are really managed. There's sort of the idea that those things are sort of addressed in these other initiatives, for example, through CEPI or the work that you're doing or other areas. But I'd have to say it's relatively fragmented. And the WHO effort to create kind of a central global discussion on the IP front through CTAP has not gone very well. And also, they have not done a very good job of distinguishing what the CTAP is compared to other things that are going on. In particular, the WHO has this COVID-19 tools accelerator, the so-called ACT accelerator, and it has a big focus on who gets what in terms of the products, and that has received far more attention and far more high-level endorsements from national governments than anything having to do with patents or intellectual property. Now, we think that the sharing of products, our shortages of products and the hoarding of products is just, it's bad, but we think it's going to be somewhat inevitable. I mean, we sometimes start by saying if Germany is 1% of the global population and they have a vaccine developed in Germany, it's unlikely that Germany is going to export 99% of the vaccine. And I think that's true not just for Germany, but certainly for the US. They've demonstrated that through President Trump's actions. And I think all countries are looking at the AMC, these preferential purchasing agreements, as a way of having some kind of guarantee that they get a certain amount of product. And, you know, I don't want to endorse that kind of activity, but we think it's just going to happen. But what we really think should not happen is the hoarding of the know-how and how to make vaccines, or the rights that you need in terms of rights and patents, data, access to cell lines, and things like that that are necessary. Now, in the U.S., where I'm located primarily, we have a national law, and we're one of the big funders, as you know, on vaccine development. The U.S. normally has a law called the Bayh-Dole Act, which sets out some rights the government has and things that we fund, including the obligation to make products available on reasonable terms, government royalty-free, and other things. However, under the Trump administration and the actions of the Congress, bipartisan action of the Congress, we've created a brand new, or I should say brand new, but a different legal authority for funding vaccines that's being used a lot right now, called Other Transactions Authority, or OTAs. Under these OTAs, the BARDA and the NIH are able to circumvent the public interest rationales on pricing and access and competition that would normally apply on a federal government grant or contract. And they've written contracts which have restricted the federal government's rights, privatized more of the research that we fund, and eliminated some of the obligations that we've had on pricing. And that's something that we've done a fair amount of work on, and we're very active in the US on. I would say for vaccines, as I'm sure people here are really deeply aware, any type of technology transfer should be begun early. I think the previous presentation was really helpful because it emphasizes the fact that the different platforms have different, they're not all in the same boat in terms of what's possible, in terms of tech transfer, in terms of manufacturing, in terms of how what the capacity is international. But we think that all the people that are funding research and development, whether it's BARDA, with the NIH, with the Canadians, the Germans, the Japanese, the Chinese, anyone that's the Italians, everyone that's working on vaccines, we think they should have, as part of their program, a commitment to and a plan for to have deep technology transfer to manufacturing. So to the extent that things are ready to roll out, there's not huge delays in the scaling up of other manufacturers, and there's just no monopolies. But now also no unnecessary time delays. That's not really happening right now, in our opinion. We think there's a high level of rhetorical support for access, but we think when it comes to issues of patents, there was an editorial in the Washington Post, I think yesterday, that was signed by, I think, 7 heads of state, including Trudeau from Canada, about how everything has to be equal globally, and that was great. And there was a statement that Gavi put out about making vaccines available everywhere. But none of these statements have really mentioned patents. Or the IP rights and the data or the know-how that are necessary. And I think it's really a bad thing that in a COVID-19, in a big pandemic, you can't actually talk about these things plainly, since they're obviously critically important. Incentives, if needed, can be delinked from the monopoly and the prices through things like market entry rewards. And this is my final slide. So what does the WHO COVID-19 access pool have to do? That would be that we'd have to, we think they should be making concrete asks to the people that are funding R&D and manufacturers and other right holders for the sufficient rights to the inventions, data, materials, and making requests for deep commitments on technology transfer. We're unhappy about the fact that we're in July and the WHO has yet to describe what it actually wants in practical terms for people to share with its pool. people barely even know about the pool. And they've had off the record conversations, but they haven't had transparent written conversations that make the governments and the funders and the companies more accountable for saying, are you in or are you out on this kind of a deal? I think they're in many respects, I think they've been intimidated by pressures or perceived or real that they think are coming from the US, Germany, the UK, the Gates Foundation, in some cases Japan, other companies, Sweden, that have taken very aggressive positions in the past in WHO negotiations on issues involving intellectual property. You want me to wind up? Is that why you're interrupting my video here?
Not interrupting. It's just giving you a message.
Okay. Well, and the last thing I will say is that I think that we're very concerned about the lack of transparency on these issues. I think in a pandemic, in a national emergency, governments should avoid secrecy on things that are critically important. We should know what is in the funding contracts. They should be transparent. They shouldn't be secret. They shouldn't be heavily redacted, and they are. And I'll just stop here. Thank you very much.
Thank you very much, James. Those are significant issues, and I hope that we will have a chance to cover them in the panel discussion. I wanted to say a few words about how COVAX is thinking about equitable access, and then we go into the panel discussion. So as, can you all hear me? Okay, so as Deborah Yersky from CEPI described in her presentation earlier today, COVAX is the vaccine pillar of Act A, the Access to COVID Tools Accelerator that a bunch of global health organizations, as well as some high-income countries supported, especially WHO and EC, European Commission sponsorship, was put together and with the goal to accelerate COVID-19 tools and equitable access to COVID-19 tools. Despite all of the nationalist headwinds that we read about and hear about every day, and James Love was also alluding to. A problem, of course, is that the whole world is affected by this pandemic right now. So every country feels like I need to take care of my citizens first. So this has been extremely complicated, bringing people together, bringing organizations together, bringing countries together. How do we raise money for equitable access? And for COVAX has now come up with the very ambitious goals that you heard about, 100 million doses by the end of 2021, and 2 billion doses for the world by the end of, 100 million doses by the end of 2020, and 2 billion doses by the end of 2021. Now, how much is that going to cost? Well, there is an investment case now, and it's going to cost $20 billion. So it's a huge amount of money, and it's very hard to raise in a very short period of time. Why do we need so much? Well, we need it for R&D. Many of these candidates are still early stage. They need to get into advanced clinical development. We need to do at-risk manufacturing, which is the only way that we can compress timelines, right? In normal vaccine development, first you will get your phase 3 data, and then you will do commercialization. Here, we have to start making the vaccine or start manufacturing the vaccine before we have phase 3 data or even phase 2 data to be able to have enough doses by the time we want them in 2020 and 2021. And many of those doses will be wasted because the vaccine might not work. So we have to pay money upfront to make vaccines without knowing whether those vaccines will work or not. And finally, we need money to procure vaccines for LMIC, for low income countries and low middle income countries. And all of that money is needed in the next few months to be able to complete the R&D for multiple candidates, manufacture hundreds of millions and billions of doses in the next 12 to 18 months, as well as to be able to deliver those vaccines. So procurement and delivery costs conservatively are estimated at $20 billion. What I would, this is a very live conversation right now, figuring out how to do this. And there are multiple scenarios that are being considered as potentially viable, including development finance funding, or charging a premium to the high income countries to subsidize the at-risk manufacturing, as well as low middle income and low income country purchase. Some of those conversations are actually happening today, and we will know by next week how much progress has been made. Let me stop there and open up for questions. And my first question will be to Dr. Yadev. I want to ask, how are you thinking about what kind of risks are involved in at-risk manufacturing? If we have phase 1 data, let's say we have phase 1 data, and investments are made, or money is given to 6 or 7 different companies, let's say, to make product at risk without knowing their clinical information. How do you think about how many should we be investing in at risk? Is there enough capacity there to do all of this? How much of this capacity is fungible? You refer to that a little bit. And how much of it is likely to get lost because the product didn't work? So lots of questions. I know you have to leave at 10, so I'm going to put them all together. Thank you.
Thanks, Anita. So I don't know if I have answers to all of the questions, and I think the meeting that you described and other things that are happening are going to get us some closer to those answers. But I think what's important to recognize is that we are doing, or we are trying to do, 2 things at risk. One is having some stock manufactured at risk after some early phase 1 data. And the second is to have manufacturing capacity ready to start production when we have results from phase 3, right? So those are 2 things which are very closely connected, but in a way, it's worth thinking about them as separate. So I think for the first one, which is building some stock at risk, I think for all candidates which are in clinical trials, depending upon the size, which means, are you talking about having 1 million doses ready or having 5 million doses ready? And that's what gives us the answer to, would we do it with all 10 candidates? Would we do it with 5 or 6? But once again, it depends upon a very good portfolio analysis. And this is something that a few colleagues and I have started working on very recently, where it's important then to understand what is the probability of success matrix looking like? And most importantly, what are the covariance and correlations between different candidates? So is it negatively correlated to the success of one? Is it positively correlated? Or it's not correlated? And that will help us answer the question of, should we invest in building 5 million doses of 6 candidates, or should we invest in doing that for all 10 or for 3? The capacity question is slightly easier in that sense, which is we don't require exclusively putting grant money out or contractual money out for building capacity. It would be a combination of grant and contractual monies, but also greater visibility and transparency of demand, which will then incentivize the companies to self-invest in capacity. So that's the part that's embedded in the advanced market commitment or the advanced purchase agreements. So through the combination of those 2, I think we will have sufficient capacity. The key question becomes the coordination mechanism, so that where we do have fungibility, how can we very quickly reconfigure a contract manufacturer's capacity, which is allocated to candidate A to candidate B? And there it's, there are 2 things that come to play. One is the contractual agreements are between the developer and the contract manufacturer are not transparent, like I think Jamie was pointing that out. The second is that there are also IP clauses in some of the cases. We've seen one example where the supplier of clinical material believes that they own a portion of the IP in the clinical material that they had co-developed with a vaccine developer. So I think that's another element to keep in mind. How do we coordinate the network? And I think that's where we look to COVAX as the group or the umbrella structure which can do this, working in collaboration or in close concert with some of the national governments who've gone ahead and made their own investments.
Thank you. Let me ask you another one, which I think will be helpful for our participants to understand. Let's say that there isn't enough vaccine manufacturing capacity, and there probably isn't to immunize everybody with 2 doses around the world in the next 2 years. How does one increase manufacturing capacity? How long does it take? What are the options there?
Yeah, so I think, again, depends upon the stage of manufacturing. So I think for bioreactor capacity, we are at a stage where for certain platforms, it can be done relatively quickly, given that we have started using these single-use bioreactors and things which are easy to bring online. For fill and finish capacity, which I at least believe we have sufficient, but in case we need more, I think it takes a slightly longer timeline to bring that up. But there might be opportunities to do. So when we think about building capacity, we think about, do we set up a greenfield plant, which means start from scratch, or do we build a brownfield plant, which is add a production suite or add a new facility or a line to an existing plant? And we certainly don't have the luxury of building greenfield plants because they will take 2 to 3 years. But with a very compressed timelines, we can have brownfield plants which can come up to start producing between 6 to 12 months time.
Thank you. Thank you very much. James Love, can I switch to you now and ask you a question related to, this is indirectly related to what you talk, but it's kind of how I think about risk mitigation in the kinds of problems that you're describing related to IP. How do you think about, for example, the global scientific community and funders are right now funding multiple candidates to get across the finish line so that there is enough, so there isn't a monopoly, right? There isn't and for many of them, it is public funding. Of course, the US is a whole different ballgame, but from in other countries, where there are what we call global access clauses, where the product has to be provided at low cost to developing countries. So how do you think about that?
Well, I think it's good when there's you're talking about the product. So when there are provisions in the funding agreements, and there are, of course. And there's been a lot announced just yesterday on this front, which was very positive. It's a good thing. I think it's interesting that the Advanced Purchase Fund is a bit of a mixed. I know that's endorsed very much. The Gavi's been very engaged in that. That the Harvard Group, Michael Kromer, the Gates Foundation, is very keen on the Advanced Purchase Fund things. The problems with those for developing countries is just it allows people that have more financial resources to stake out bigger claims. In theory, I know there's policies designed to mitigate that. So I think those are positive. I think that the lack of transparency in all of these things is problematic for us. I mean, I know that I'm not criticizing the Gavi policies on this, but I'm just sort of saying at the national government level. I think partly the governments are like the Oxford contract, for example, with AstraZeneca, which is quite important. Oxford has been willing to give off-the-record briefings on some of the details to some of the NGOs that are making inquiries, but they won't show them anything in writing, and everything is kind of like in some kind of Zoom call that isn't public. And I think that everything about the quantities, the prices, and the IP should be public, and we should be able to have adult conversations about what's going on in this area. But the reasons why they're not public is in some countries I think is like a national security interest. I think the UK government probably intervened in the Oxford case because they just sort of see it as a matter of national security for the UK to have some kind of preferential access to the products. And of course everyone's thinking the same way. What the problem for everyone is that no one knows who's going to cross the finish line first, right? As you mentioned, that there's lots of products, there's lots of vaccines under development. To the extent you get everyone to commit to putting their cards on the table, and everyone committing to doing the kind of brownfield kind of technology transfer where it's possible, if depending on the platform, it just seemed like it would be in everyone's interest to do that. You just hedge your bets. Is it going to be the Chinese? Is it going to be the Italians? Is it going to be the Germans, the UK, the US, the Canadians? I mean, who knows, right? So unfortunately, people are just not transparent because they don't want to expose whatever preferential agreements that they have, and they're also still in some ways. And certainly the feeling of WHO is Germany's going this way is there's some kind of industrial policy thing going on where they think they can make money off the vaccine, and that's really, I think, unfortunate.
Okay, thank you. I think that's all we'll have time for. There are other questions in the question and answer. If you, Dr. Yadav and Jimmy Love, if you have time to address them later on, that would be great. We would not have time to cover them today. I think most of those were covered in your responses anyway. I can very much agree that our current we will only solve this problem by all of us working together, right? And if we can't do that, this is going to be a very hard problem. It'll be impossible to end this pandemic, and so it is a call to action from GLOPIDAR that countries and scientists and manufacturers, everybody needs to work together to solve this together. Okay, let me, now we are running 1 minute late, and I'm going to pass it on to my colleague and co-chair, Dr. Deborah Yaski, to close out with the last and final session of today on clinical trials and regulatory consideration. So over to Dr. Yaski. Thank you.
Thanks, Anita. And I have to apologize for my technical difficulties at the beginning of the meeting here. It was sort of maybe like a drop the mic scenario when I was done. But I'm very glad to host this session of clinical trials and regulatory considerations. And so in the interest of time, I will give the floor over to a colleague of mine at CEPI, Amal Chaudhry. He is currently the clinical development lead at CEPI in London. And before CEPI, he was at the Serum Institute in India as a medical and operations lead for clinical trials for vaccines. Amal?
Thanks, Deborah. I hope my voice is clear. So thank you for the introduction, Deborah, again, and opportunity. Thank you for the opportunity the organizers has given me today to present some thoughts on the consideration for efficacy endpoints in COVID-19 vaccine clinical trials. So I'll focus my talk on 4 broad elements, starting with the preference of symptomatic COVID-19 as a primary outcome of interest, as opposed to SARS-CoV-2 infections. And I think this comes from our historical learnings with other respiratory virus vaccines, which tend to impact more on the severe disease, mortality, and ICU hospital admissions, as opposed to, say, mild disease or asymptomatic infections. Next, I want to touch upon the choice of primary endpoint in terms of symptomatic COVID disease, but which severe, like all severity COVID-19, or just focusing on moderate, severe, or critical COVID-19. Third point about the positive predictive value of the COVID-19 associated symptoms and their importance in triggering nasopharyngeal swabbing for PCRs during trials. And finally, a bit about asymptomatic infection and its importance as a vaccine efficacy endpoint in the context of especially the reliability of available assays. So if you could go to the next slide, please. So as we heard previously as well today that the respiratory viruses or the COVID-19 vaccines are likely to behave similar to other respiratory virus vaccines such as influenza or rotavirus, the mucosal virus vaccine, in the sense that they may not block the infection fully, and the recipients would still have some mild residual symptoms, but it will have more impact on severe disease and progression of the disease. We have seen at least one animal study from one of the vaccine candidates, which showed that challenged non-human primates were protected from severe disease despite showing milder symptoms. But of course, this is just one animal study, and we should always be cautious when trying to interpret these results and translating into humans. There is, however, a consensus evolving around globe among various stakeholders such as WHO and even the other trials that are looking for efficacy as primary endpoint in choosing COVID-19, symptomatic COVID-19 as the primary endpoint as opposed to SARS-CoV-2 infection when looking for COVID-19 vaccine efficacy. So if you could go to the next slide, please. There is, however, still some debate on what does a symptomatic COVID-19 as a primary endpoint looks like in a clinical trial protocol, for example. So just a couple of scenarios here I'm presenting that there could be more options here, but to start with, as a comparison, one way of doing it is any severity COVID-19, which would be preferable in low incidence settings where, for example, the peak of the epidemic has probably passed, and one can afford to enroll younger and less vulnerable trial population, since they would present with the entire spectrum of severity and help in gathering the cases faster. But there is a risk of including vaccine attenuated disease, like if the vaccine only prevents disease progression, then the vaccine recipient should still present with milder disease, and that gets counted in your primary efficacy analysis, which may impact the vaccine efficacy estimates. On the other hand, if one chooses to go with what I'm calling here as moderate to critical COVID-19 as defined by WHO, which requires the presence of pneumonia and signs and symptoms of pneumonia. And this could allow, in more useful in, say, high incidence settings, and you could enrich your populations with the trial populations with the population that is more vulnerable to this kind of severe disease, such as older and other maybe comorbidities, morbidities. Sorry. This may also allow the developer to exclude cases of vaccine attenuation, thereby again maybe hastening or speeding up the achievement of vaccine efficacy estimates. If you could go to the next slide, please. This slide and the slide after this, what I'm trying to present is the diversity that again exists in terms of defining the severity of COVID, as well as COVID as a case itself, and what constitutes the signs and symptoms that go into defining this symptomatic COVID per se. So if you go to the next slide, please. And these are the symptoms of COVID. And if you see, there are various guidances available here. Of course, the US CDC and European CDC focuses on the surveillance for surveillance purposes, but the symptoms, signs and symptoms that are associated with COVID are variable, and various organizations have included, and which is not surprising actually. I mean, we are learning about this disease as we go along, and the information is still evolving. But what this creates is a dilemma for a vaccine developer during the trial conduct in trying to define their case definition. And there may be a case, as I've made a point here on the slide, that there may be a case of considering a clinical endpoint adjudication committee as well in the efficacy trials. By the way, just to clarify, when I say COVID-19 here, it may not be explicit, but what I'm implying is virologically confirmed COVID-19 via RT-PCR. So if we go to the next slide, please. Similar to this, there's a dilemma of when to trigger case workup, or that is nasopharyngeal swabbing and PCR in a clinical trial, whether if a participant shows any of the symptoms listed above, or you stick to more specific indicative COVID-19 symptoms. For example, maybe a fever, cough, dyspnea, or anosmia, agasia. In the former case, there is a likelihood that you might overburden or overload your testing capacities, especially if the trial is happening in very high incidence settings and where subjects turn up with febrile episodes every other month, and if you coincide with, say, flu season, and then you get a lot of flu-like symptoms, which then need to be tested for PCR. On the other hand, if you choose only a stricter or a higher threshold and go for PCR swabbing only in case of indicative symptoms, there is a chance that you may miss out on few atypical cases, which could delay the achievement of vaccine efficacy endpoint. What could help here is an exercise where you try to determine the positive predictive value of especially non-specific symptoms when they are seen in isolation without the concurrent specific COVID-19 symptoms. So if you go to the next slide, please. And to wrap up, the final point about infection, especially asymptomatic infection, which we heard earlier could be very important in the context of preventing transmission. But when assessing asymptomatic infection in a trial, it could be very challenging that you may need weekly swabbing and PCRs, which is not just costly, but is further complicated by the specificity of the test, where if it reduces by even 0.1%, for example, you would get hundreds of false positive results, which again can skew your vaccine efficacy estimates. An alternative way is to look for antibodies against an antigen which is not part of the vaccine. For example, if it is an anti-spike vaccine, then you maybe do anti-N antibodies at specific time intervals. But this is also complicated by a couple of things in terms of the sensitivity. If it is low, then it could bias it towards the null hypothesis. Then again, we are not really sure about the persistence of anti-N antibodies and there are some reports which say that it could be less sensitive in mild and asymptomatic cases. So if you go to the last slide after this, which I just want to highlight those points again, and maybe this could be a kickstart the discussion around these going ahead. So thank you. That's it. Deborah, are you on mute?
Okay, it looks like Deborah has had an inconsistent connection for a while, so it's Charu here, and I'm going to introduce the next speaker, who is Gagandeep Kang from CEPI, India. She's also from the Christian Medical College in India, and she's going to talk to us about the COVID-19 vaccine development requirements for clinical trial sites and regulatory considerations in LMIC. So Gagandeep, I'll hand it over to you.
Thank you very much, Charu. Can we have the next slide, please? So if we take a look at a map of clinicaltrials.gov and the trials that are registered, you can see that some parts of the world have many more trials than others. Now, clinicaltrials.gov is not the only primary clinical trials registry, but it is telling that places in the world that have lower clinical capacity are the ones that do less clinical research. A while ago, it was thought that because developing countries, LMICs, had access to very large populations, they would be great places to do clinical trials. And there were a number of clinical research organizations, researchers from academic backgrounds and from industry who came to sites that had national regulatory authorities that were reasonably placed, or at least places where WHO recognized the NRA and set up to do trials there. Next slide, please. There was a ballooning of trials that happened, and there was a fallout from that. Basically, because in a rush to recruit patients quickly and to cut costs as much as possible, there was a fair amount of exploitation of patient populations. There was a lack of awareness of the rights of participants. There were safety issues in trials with adverse events not being reported. There were other ethical challenges, such as the poor quality of informed consent or of review processes. Patients essentially were not protected, and if they had trial-related injuries, then issues of compensation were very rarely settled, because the whole process of getting insurance and then calculating how much compensation was to be paid had not really been worked out very well. And of course, there was a lack of post-trial access to successfully developed drugs and vaccines. This led to a lot of discussion, both in country and among partners, to say that this was a situation that could not continue, and there has been a lot of excellent guidance that has been put out since then, focusing on trying to improve this situation. The next slide, please. When we look at low and middle income countries, there are also challenges of facilities, of sites, of processes, of not having people who are trained in the same clinical research environment as can be found in more industrialized countries. There's also a lack of investigator-initiated trials because there isn't expertise for design and conduct of such studies. Laboratory support can be an issue, and frequently when trials come into countries, when the problems are huge, you can have competing research priorities. And the funding for execution, particularly when the funding is derived from in-country resources, can be quite insufficient to address deficiencies. That said, there are in South Asia, in Africa, truly outstanding clinical trial sites, and we are going to hear from a couple of people who have tremendous experience with conducting clinical research in Brazil and in South Africa just after this. The next slide, please. Now, if we look at the kinds of systems that we are dealing with in low and middle income countries, there can sometimes be a mix of public and private services where access to healthcare is a huge issue, and many patients need to pay out of pocket. There are limited government services. They can, when you need to access care for certain kinds of conditions, they are either not available or are very expensive. In these kinds of settings, you also have multiple systems of medicine, many which rely on culture or religion, which are traditional treatments, which can also make the conduct of clinical research quite difficult. There is a lack of patient education and support, and healthcare staff are not familiar with trials. All of it can raise the bar for trying to do trials in developing countries. Next slide, please. Now, when we come to regulatory challenges, many regulators in low and middle income countries are reliant on having acquired expertise in looking at products that have been made elsewhere before, that have been through a regulatory pathway elsewhere. So when it comes to first-in-human studies, they are a little nervous sometimes about approaching these kinds of studies, and the requirements for them are not uniform. There is a complexity in the approvals process. Frequently, you have multiple agencies that overlap and have different processes. So then anybody wanting to do a trial, particularly a regulatory trial, has to run around to get many kinds of approvals, and it can become quite circular. For innovations, it becomes challenging, and some countries, this is changing, particularly for SARS-CoV-2, but accelerated approvals for innovative products are sometimes hard to find. The consistency and the quality of the review can vary, and it would be outstanding if we had regulators that had timelines and performance management so that there was responsibility on them to be able to deliver a quality review. Review committees change a lot, and having the same review committee through the development cycle of a product would be a huge advantage to those seeking to have new drugs or vaccines tested in low and middle income countries. Pre-submission discussions are now being arranged in Africa and in Asia, and these have led to some easing of the path for getting approvals for trials. Looking at collaborations really between the regulator, government, and industry and academics would be a good way to think through how we can counter the risk aversion that reviewers currently have for testing any product where the safety profile has not been defined elsewhere. And there is an approach now to look at injury compensation requirements and at safety monitoring. So while I paint a pretty bleak picture of what has happened in the last 20 years, this has been the basis for a lot of change that has been driven by governments, by regulators, by the World Health Organization, both in Asia and Africa, that have brought regulators together from different countries to establish platforms so that clinical research can be better conducted. Next slide, please. This is just my last slide, and this is a clinical trials toolkit that was developed in India. The idea here was to support both industry and academic researchers with all the steps that are required for clinical trials. This is something that is aligned with the regulatory pathway in the country and makes it easier for people to do trials in India. This is built off a similar toolkit that's available in the UK, and we could be thinking about having similar products available in other places as well. So I'll end there and leave you to the next two talks that I'm looking forward to as well.
Thank you so much, Gagandeep. So we're trying to stabilize Deborah's connection, and hopefully she will join us for the panel discussion. In the meantime, I'll do the easy job of introducing the next two speakers. The next one is Ricardo Palacio, and he is currently leads the clinical development of immunobiological products, mainly vaccines, as the clinical research medical director at Instituto Butantan in Sao Paulo, Brazil. And he will be talking to us about the experience of setting up a trial in Brazil. So Ricardo, please go ahead.
So thank you very much for the invitation. So next slide, please. So first of all, let me introduce my institution. So we are an institution that is kind of an old-fashioned institute-based model. So it was a research institution, a public institution that also has a manufacturing plant. We actually are one of the largest manufacturers of immunobiologicals, serum and vaccines. We produced 70 million doses of flu vaccines this year. For the public health system of Brazil. And we work in several partnerships for vaccine development with private institutions, big pharma, and with public institutions as well. Next, please. So whenever we started partnership, and this was not different for COVID-19 vaccine development, we assess, of course, non-clinical and clinical aspect, but we also assess the feasibility of tech transfer for local production and how we can agree in future distribution. So we usually set up different kind of agreements according to this assessment. So agreements on non-clinical or clinical development, agreements in product development. Sometimes it's necessary to have some further development of the product. Please ensure of a distribution agreement and the tech transfer agreement. We are committed as a state policy in Brazil to have the self-sufficiency for the country, and in the latest years we are more focused also to supply regional markets. Next slide, please. So in the assessment, to talk in particular of this vaccine with Sinovac, that is inactivated vaccine, I will point out two key articles that allow us to move forward in this process. The first one is kind of a old article. This is from 1990, and it was an human infection model with a common cold, a coronavirus, the 229E. And in this article, and I will want to draw your attention to the lower graph, and the black dots are the ones that have low antibodies at the beginning of the experiment, and the white dots are the ones that has high antibody levels at the beginning of the experiment, and they were challenged with this coronavirus. And as you can see, the ones with the black dots, they actually were challenged, they were infected, and they have an increase of the antibodies, and then a decay. And in the other ones with high levels, they were unable to infect with this experimental model. And the other thing that we consider is that Sinovac, during the SARS at the beginning of the century, they create a vaccine, an inactivated vaccine, and they were able to demonstrate with this technology in 2004 in a phase 1 trial that they create a vaccine with a good level of neutralizing antibodies that are the ones that in the other graph is shown is the same kind, and we realized that probably this could be a good strategy. In addition, the inactivated technology is a very old technology, very reliable in producing antibodies, and this is something that we can easily incorporate. Next slide, please. So the first agreement that we have is a clinical development agreement between the two companies. So we have some studies that will be in charge of Sinovac, and some studies that will be in charge of Butantan. So our first study in this development is a phase 3 study with healthcare professionals with near 9,000 participants. Next slide, please. So there are some key aspects that some of you have discussed, but are very critical in the protocol design. And the ones that we are pioneering these phase 3 designs, we are having troubles in defining which definition, for example, case definition, as and Dr. Amol just mentioned, how to make the diagnostic of the sampling and age limits and previous exposure and so on. So several discussions with the regulatory agency. Next slide, please. So let me show you very quickly. This is our timeline since the signature of the agreement in 11th of June until the first participant in that is scheduled next Monday. So you can see from the submission, the formal submission of the IND dossier and to the first patient in, we will have near 1 month distance. Next slide, please. And this is just to show you some of the states that we choose and epidemiological analysis. So we choose in several states in the country. Next. And of course, we have to work in parallel to all these epidemiological assessment, work with the regulatory approvals, ethical approvals, also in site readiness, in meaning site selection, vendor selection, and establishing the gaps for operational assessment, legal aspects, training, and so on. Next slide, please. So this is the list of the sites. I want to thank all of them, all the teams in the clinical sites. And next, that is the last one, please. And of course, I want to add thanks in advance to the volunteers that will be really the key for having an answer in this first vaccine. And we expect to pave the way for the vaccines that are coming. So thank you very much.
Thank you very much, Ricardo. That was a really nice overview. And we'll hear of a similar experience now that Shabir Emadi is going to tell us from South Africa. And Shabir is a professor of vaccinology at the University of Witwatersrand, Johannesburg, South Africa. And he's a co-director of the African Leadership Initiative of Vaccinology Expertise. And he's the principal investigator of the South African COVID-19 vaccine trial. So Shabir, we look forward to hearing your experience of setting up a trial in South Africa.
So good day, everyone, and thank you for providing me the opportunity. So the study that I'm going to be presenting on that's currently enrolling, in fact, I'm fortunate in that Sarah Gilbert has already introduced the vaccine. And there's a chimp adeno Oxford novel coronavirus vaccine. And the study that we're doing in South Africa is a phase 1, phase 2 study looking at immunogenicity and safety in a limited number of individuals with HIV, as well as looking at safety, immunogenicity, and efficacy in adults without HIV. So the sponsor vaccine developer is obviously University of Oxford, but the study is actually funded by the South African Medical Research Council and the Bill and Melinda Gates Foundation. The arrangement with Oxford to actually undertake the study in South Africa was done prior to the University of Oxford entering into an agreement with AstraZeneca. And it was really the South African investigators reaching out to the University of Oxford to see whether they would be interested in at least providing vaccine for such a study to be conducted in South Africa. So the study is currently being done in one of the 9 provinces, which is Gauteng, which is the most densely populated province in South Africa, and will be expanding to a second province in the next week. Next slide, please. So it's got 3 different arms. In the first group, it's basically looking at people without HIV, looking at immune responses and safety to a 2-dose schedule. The second group, which is a larger expanded immunogenicity safety group, also has a vaccine efficacy component built into it. It's an adaptive study design insofar as at the start of the study, we weren't sure whether we were planning on using a single dose or 2 doses of vaccine for group 2, which is the efficacy group. But based on the most recent data, it appears that we will be using a 2-dose schedule. And then the third group is the group with HIV. And the reason why this is especially important to include in South Africa is that there's quite a strong demand from civil society to ensure inclusiveness of people living with HIV in terms of the clinical development of vaccines as well as other therapeutics. So there isn't a leg in terms of deriving the necessary data to inform its use in that particular group. In terms of the efficacy outcome, the main endpoint is basically looking at all severity of COVID-19 disease in individuals that are COVID-19 naive at the time of randomization. And that is to be both seronegative as well as testing negative for the virus within 96 hours preceding randomization. And this is actually something that we needed to sort of amend the protocol for, as I'll show you in the next slide to follow. Next slide. So Cherry sort of raised the issue in terms of some of the regulatory hurdles when conducting studies in low middle income countries. And South Africa is relatively fortunate in that we've got a relatively mature regulatory framework. But even then, previously prior to COVID-19, it would usually take anything between 4 to 6 months for a study to be approved by the regulatory authority, which is known as SAPRA, as well as by the ethics committee, which usually take 2 to 3 months. We were fortunate in that both the Ethics Committee as well as SAPRA, the regulatory authority, had put into place measures to ensure an expedited timeline in terms of approval, in terms of review as well as approval of studies related to COVID-19 therapeutics as well as vaccines. And consequently, we were able to condense that time period from 4 to 6 months to 4 to 6 weeks. In addition to which amendments are basically being reviewed over a period of 2 to 5 working days, which has really assisted the study. Next slide, please. So we've started a study in South Africa at a time when we're actually very much on an upward trajectory in terms of this current wave of the outbreak. As you can see, we have not as yet peaked, but compared to many of the European countries, they're pretty much waning in terms of the epidemic. So that would appear as an advantage, but at the same time, it's come with some challenges. Next slide. And one of the major challenges is at the time of the start of the study, the plan was to test for infection at the time of randomization. And what emerged after having enrolled the first 100 subjects is that roughly about 20% of participants that were asymptomatic, that were being screened at the time of randomization, ended up testing positive for the virus. Either a positive test or an indeterminate test. And many of these indeterminate tests ended up being positive because it appears they were being caught at an early stage of infection, or some of them might actually have been recovering from infection that preceded their enrollment. So that required sort of a change to the protocol and an amendment, which now requires for a screening to be done for active infection at least 96 hours before the person is randomized. And even with that implementation, we find that with a screening visit, roughly about 20% of volunteers that are now being screened are either positive at the time of enrollment or have a result which is indeterminate. But like I said, many of those indeterminate results are turning out to be positive on subsequent testing. Next slide. So the current timelines is that we're planning to complete enrollment of 2,000 participants by August 2020. And the efficacy readout is looking for 60% efficacy with a lower bound of greater than 0%. So we need to accrue 42 cases and at least 14 days after the first dose of vaccine. And the projection is we probably would reach that endpoint number roughly by about the end of November, December, because of the large force of infection currently underway in the country. Next slide. One of the issues that we've had to deal with was some sort of a protest with regard to conducting studies in South Africa because of some unfortunate remarks from 2 French scientists related to BCG vaccines should be, in terms of its possible protection against COVID-19, that those sort of studies should be done in Africa, and I think most people are aware of that. But by and large, we've had a very favorable response in terms of enrollment, and this has been assisted by, as an example, the Dean of the Faculty of Health Sciences at the university, also volunteering to participate in the study. Next slide. So thank you for your attention.
So thank you so much, Shabir. So excellent overview by the 2 speakers regarding their experiences and where they're at in terms of setting up these trials. I think we have Deborah's connection stabilized. So Deborah, are you able to take over? And I can start off with posing the question, which Marian Koopmans has put in the chat box. And, you know, I think many of us are questioning that, regarding the possibility of multinational trials, whether they're in LMICs or across countries. As we know, over the past 6 months, the pandemic has traveled across the world. And by the time the vaccines are going to be, looking at the efficacy, for example, many of the vaccines that are initiated in Oxford or in other places, and certainly in Canada, we are going to struggle to be able to do any kind of efficacy. South Africa and Brazil, not that it, but are in a position to test that. But 6 months from now, are we going to be able to actually look at that? So I guess I'm going to start by asking Gagandeep to weigh in on that in terms of what would be the mechanism to move forward to see the efficacy.
In terms of looking at multinational studies, while it's true that there has been a focus on high and middle income countries so far, There are many low middle income countries that are willing to participate in trials. WHO has the Solidarity 3 protocol being developed, and they have asked for expressions of interest from multiple trial sites and have many sites that have signed up to participate in trials. Many regulators are also willing to review such studies and conduct them. In addition, the US is using its clinical research network sites that have been supported through the National Institutes of Health. And their intention is that many of the vaccines that they are testing might be tested offshore as well. So I think it's very likely that we are going to have multi-country studies. And obviously the goal right now is to do multiple vaccines being compared in the same trial. One of the biggest worries, of course, is when you start getting into phase 3 trials, do you have enough sites and do you have enough patients to be able to test all of the candidates that are in the pipeline at the moment?
Okay, so I guess at this point, I'm going to ask Deborah because, as many of you know, her specialty is regulatory aspects. So Deborah, do you want to comment and then maybe take over the questions?
Sure, thank you. And thank you, Cherry, for that response. And again, sorry for the technical difficulties today. There are a lot of things going on. I'll just make just a couple comments about, regulatory and what was, said before. Regulators across the globe are, learning as much as we all are at the same time. And so, and they're trying to be as flexible as possible within the constraints of the regulations of their region and the like. And so, and I can speak as far as, FDA released guidance a week or so ago, which on the onset seemed very, conservative and sort of maybe traditional in the sense. However, one needs to remember that guidance is just that. It's recommendations, and it really comes down to the traditional relationship that the agency or any regulator has with the developer. And when it comes to that regulator with data and justification and rationale, if it doesn't mirror what is spelled out in any type of guidance. And so that being said, I think that there's enough room and tools in the regulatory toolbox at this stage, to get where we need to go. The conundrum is just that, though, that for large scale phase 3 efficacy trials, that because of the waning numbers in areas, across the globe, that is a conundrum. And I think that I know that various groups, including WHO and vaccine cluster meetings and organizations such as ICMRA are trying to look at these global regulatory issues. So I'll ask the next question is for Sabir, is did you need to have enough space for social distancing, and did that slow the enrollment process?
Yeah, so we've needed to set up sort of makeshift facilities to ensure that we can maintain physical distancing, in addition to which we've sort of got a roster that participants need to adhere to in terms of coming in. So we staggered throughout the day. So it's worked reasonably well. I don't think it's been an impediment in terms of the rate of enrollment.
Okay, thank you. I don't have any other questions from the Q&A online. I guess maybe if I can just ask a question is, and to the group, and I'll start with Cherry. How can we ensure that the global capacity to test the most promising candidates in clinical trials are sufficiently powered to provide the highest possible level of evidence on safety and efficacy?
I think this is where global consultations are very helpful, where having regulators talk to each other is useful. What tends to happen if regulators are working on their own and working with companies that they've worked with before, they adhere to local standards and not necessarily international ones. The fact that the FDA has now put out guidance is very useful. There is an estimate of the number, the protocol, for example, for the Moderna site being posted and the registration in clinicaltrials.gov that gives you the size of the trial that needs to be done in phase 3 is particularly valuable, because you're aiming for a point estimate of 50% efficacy at least, and that's going to require 30,000 participants. That's a reality check, I think, for many regulators, particularly in LMICs. And if we do these studies that become multinational studies, all the regulators are going to have to realize that these studies need to be large so that they are sufficiently powered. I think that's a realization that is coming to them, and the more consultations that we have like this, the more interactions there are, the better it will be.
Thank you. Ricardo or the other, Amal or Sabir, would you want to comment?
Yeah, I mean, I agree with Cherry. Obviously, the big issue is how this pandemic is going to unfold moving forward, and whether we can anticipate additional waves of the outbreak, which seems to be possibly the case. But I guess it's all an issue of timing. So as an example, in the South African study where we were assuming an attack rate as low as 3.5%, we needed 2,000 participants to get the vaccine readout of 60%. So it really depends on how the pandemic unfolds over the next few months, which will determine the sample size and how effective we would be and how quickly we would be able to get the result in terms of an efficacy readout from these studies.
Thank you. I would like to comment something that is, if any manufacturer, for example, of inactivated vaccine wants to do their own phase 3 trial, probably it could be a waste of resources. So we need to establish probably families of vaccines and establish immunomarkers in order that they can copy or they can use these immunomarkers to have a surrogate to get the approval and probably make some post-licensure commitments, rather than every single manufacturer trying to make a big mega trial because that could be useless. So, for example, the difference in the technology probably are not so significant between the adenovirus family, between the inactivated vaccines family, between the subunit family. So probably it would be smarter if the institutions like CEPI, WHO, and all the COVAX can foster this kind of agreements between the companies in order to truly share this kind of correlates of protections and share the information and the procedures to avoid doing the same trial and again and again and again, because that is useless. That's my point of view.
Yeah, I guess, I mean, just to chip in here, Ricardo raises a very important point, and I think we heard about the constraints and the capacities which are very limited. When there have also been thoughts, I think this was put up earlier also, whether we could have vaccines which are aligned in timelines, and could they be merged into one trials, for example. I mean, instead of blocking the capacities of sites, so maybe sequentially, and if as you know, Deborah, I mean, COVAX, for example, it's at the heart of COVAX in the sense trying to achieve this kind of global collaborations and efficient use of resources, which we know are very, very limited. So yeah, there would be a requirement of a lot of out of the box thinking for these cases.
Thank you all. I also wanted to just let the participants also know that within COVAX, and I just lightly touched over it, but just because a vaccine might not be in the COVAX structure, we have 3 SWAT teams that we have clinical enabling sciences and manufacturing, as well as a regulatory advisory group that will tackle product agnostic issues. And I can see where, and the regulatory advisory group spans the 3 SWAT teams, and issues will come up from the vaccine product groups. That's the traditional relationship that that we have with shepherding the regulatory and the development of vaccines. But just to say that issues like this, resource saving and not recreating the wheel, and talking about potentially surrogate endpoints would be something that because specifically in the FDA guidance it said that they're not seeking accelerated approval using surrogate endpoints or correlates of protection. However, I mean, we're all learning at the same time. So these types of engagements through COVAX can be utilized to help the whole field. And we're still looking how to, communicate those interactions that we have within, the SWAT teams and what the regulatory advisory group will opine on by having, open door workshops that's available for all vaccine manufacturers, just not the vaccines that are in the COVAX structure. So I just wanted to let participants know that as well. Are there any other questions? Any other? We're coming up to time anyway, and so I think that it's probably time to wrap up. But just to give my panel members anything else that you think. If there are any remaining gaps that the vaccine developer landscape and/or GLOPIT-R members can need to know.
And just to add, Deborah, I mean, in terms of again coming back to the site capacity thing and how we can, I guess there could also be a role that epidemiology plays. I mean, and prospectively looking at how of course nobody can predict how the outbreak will move, but at least learning from the previous experiences, and then that kind of activity in trying to predict, not, I shouldn't say predict, but how the outbreak will move, and based on that doing the site selections or doing your trial, going to a particular region or a site is could be a way of doing a very informed or a calculated guess or trying to efficiently use again the sites or the resources. Yeah.
May I add something? That is, when this kind of organization, a big organization, no matter which one it is, a big pharma or CEPI or WHO, whatever, is approaching individual clinical sites to join those trials to come, there is always a concern, and this is something that should be assured, is that in the public, there is a question mark whether this vaccine that is being tested in my country could be accessible to the population of the country. So this is something that should be addressed, in particular when these kind of organizations are looking at individual clinical sites, because this question is arising in the public. And we saw that in Brazil recently. We have these two approaches, the first one from Oxford that came and in this regard, and it came with one individual clinical site, and only later on, like more or less 1 month later, there is an agreement with the government, with the federal government. In our case, we set up the agreement in both sides. But this is a question mark, for example, for other countries I would mention in Latin America, Peru, Chile, that are being targeted for future clinical trials. For example, the network that has been established by the US government using the HVTN, HPTN sites, that they close the doors to other manufacturers, but they don't warrant that the vaccine would be accessible for the population. So this is something that probably should be addressed in some way, and an individual clinical site has no power to negotiate that.
Great, thank you. I think we're going to have to close, and I just will just wrap up for a couple of comments going forward. I mean, this has been a great first start in talking about, vaccines. And as I said earlier in my earlier remarks, there's a lot of information and a lot of things happening. And going forward, we are all focused on one goal. And I think that, there's much more to learn, much more to share. But I think that when we do this together in an end-to-end approach with transparency as much as possible, there are some definitely some hurdles there to work out. But I think that what we're learning in this pandemic is that there were a lot of gaps, across the international space. And I think that there will be a great lessons learned and after action after this pandemic wanes, and we get, not one vaccine, but multiple vaccines out to those that need it. And so I turn it over to Yazdin for any other final remarks. Thank you.
Thanks a lot, Deborah. And I wanted to thank all the panelists, all the speakers for this great session actually on vaccine. I think it was very useful for the other researchers who are on the field to listen to all what you have done and congratulate you for all the efforts. Although the vaccine will be something that we will hear for another months and months, I think. So thanks again to everyone. Just also to follow up to say that we will continue with this Synergies meeting. So tomorrow at the same time, we have the therapeutics. As you know, on Monday we have transmission. On Tuesday we have social sciences, and then we will have on Thursday, July 23rd, a summary of all the session and the discussion panel. And thanks to everyone. Of course, you don't need to come to many of them, but it will be great session. So you are invited, and I think that you can come in if you want to leave, but I think that it will be very important, all these sessions, and to have you all around will be very and extremely productive. And just to finish, I have 3 minutes left. I really wanted to thank for all the work done, in addition to panelists and to, of course, speakers. So first of all, Charu, who had done a great job for actually moderating, supervising, and everything, and all the team from Canada, so Geneviève, Daniela, everyone from Fondation Mérieux, from Wellcome Trust, from European Commission, everyone has made a huge job around all this to make it happen. So thanks a lot. Charu, the final word is yours.
Thank you, Yazdin. So yes, thank you to all the speakers and for really interesting discussions. I was watching, and at our peak attendance, we had over 200 people participating today. The goal of these meetings is really to bring people from across the world to discuss on where we are in terms of trying to end this pandemic. So thank you everyone for all your discussions, participation, excellent questions, and lots of discussions. And we will see you tomorrow morning at the therapeutic session. Have a good day.
Be careful. It's morning for her. It's afternoon for us.
Oh yeah, tomorrow morning, afternoon, evening. I know we are covering globe to globe.
Yeah, no, I'm just kidding. Thank you. So see you tomorrow.
Thanks very
Bye.
much.
Thank you. Bye.